those present with us online. Without much ado, I invite Dr. Jitendra Kumar, MD Bairak, to kickstart the day and extend a warm welcome to the gathering. So may I request our esteemed speakers to join us on the dais, please. Dr. Lata Kapoor, Dr. Anand Kumar, Professor Gopal Nath, Dr. Bhabatosh Das, Dr. Aradhana Vipra, Professor Urmi Bajpai, please. So I again request MD Bairak, Dr. Jitendra Kumar, sir, to please extend a warm welcome to everyone.
Honorable panelists of this session, scientists from Department of Biotechnology, my colleagues from BIRAC, scientists and other dignitaries from other organizations. I welcome you all to today's World AMR Awareness Week. This is very special because today we are also launching, actually it's not launching, it's a soft launch we can say, celebrating scientific completion of first indigenously developed antibiotic against MDR isolates, Nafithromycin, which is also called Miqnaf. Now this is one of the milestones for our country where we are launching a drug that has come out indigenously. So we have supported, BIRAC has supported, DBT has supported, and the company also is indigenously, they have been involved in developing this drug for a long time. We had supported a clinical phase 3 trial for this drug. I think the phase 1 and phase 2 was done in Europe and US. And phase 3 was in India, and we had supported some 8 crores of funds through our BIPP scheme, Biotechnology Industry Partnership Program. It was not only the funding, and as you know, our scientific technical committee, they interact with the innovators very often. Milestones, etc., everything is evaluated, and in a way this is a very good mentorship also to the innovators. So this is a funding program as well as the mentorship, the guiding. So we were closely involved and engaged with the team who developed this drug. As you know, multi-drug resistance is a huge problem in India. Around 2.5 million deaths occur every year through this multi-drug resistant bacteria or pathogens. And out of this 2.5 million, 6 lakhs death happens in India. And apart from that, there are a huge number of population, which is, I think, under one estimate, every year 19 lakhs people in India only suffer from, multi-drug resistant hospital acquired infections. The current line of the treatment, which is the azithromycin and the stage 4, as we say, the drug for latest this thing is not very effective because the pathogen develop the resistance. At least in 30-40% cases we see the resistance occurring. I think we have a eminent panel of experts to shed more light on the scientific aspects of this. So I will not go through the scientific and technical aspects of this session. But the most important thing that we have to note is the company has developed this and is launching its product. The scientific committee of CDSCO has already approved it. Now it is just a formality. CDSCO will approve and it will be in the market. Around 400 to 500 crores estimate, market capitalization can happen. There is a huge market here, and the company is already in talks with so many other markets across globe to commercialize it. So it's a wonder drug, I can say, and we must celebrate it through the efforts of government. This has come up. So with this, I once again welcome you all for this session, as well as the product launch, the soft launch with our Honorable Minister. And specifically, I would like to welcome Dr. Lata Kapoor, Additional Director NCDC, Dr. Anand Kumar, Co-founder and CEO Bugworks Research India Private Limited, Bangalore, Professor Gopal Nath, Department of Microbiology, BHU, Dr. Bhabutosh Das, Professor, BRIC THSTI, Dr. Aradhana Vipra, Head of Microbiology, Professor Urmi Bajpayee, Department of Biomedical Sciences, AMDC, University of Delhi. So welcome you all. I think this will be very, very enriching and enlightening session, because some of the most important personalities in the field, both from academia and industry, are here. So all the best wishes for this session and the launch. Thank you so much.
Thank you, sir. So innovation is the spark that ignites progress. In the face of AMR, innovation is not just an option, but a necessity. To discuss some of these innovative approaches being developed to combat AMR, we are honored to have amongst us some stalwarts in the field. Before we begin the session and invite the first speaker, it's a humble request to the speakers to please adhere to the time. So our first speaker for the day is Dr. Lata Kapoor, Additional Director and Head of Center for Bacterial Diseases and Drug Resistance and AMR Program Unit at the National Center for Disease Control. Ma'am will be speaking on innovations required to contain AMR in India.
Thank you. Thank you so much. First of all, I would like to thank the organizers for having me here. Thank you so much. This is an area of great interest to public health. I am from Public Health Institution, National Center for Disease Control. And I think these forums are so wonderful for us to prioritize research, which is very tapered for our country. So I would like to show you the first slide, which is on what is AMR. And this is a slide which I show in most forums, excepting in a forum where only microbiologists are there. So I would like to share with you that if you look at the yellow dots there, the yellow dots are the sensitive bacteria. In any clone of a bacteria, those are the sensitive bacteria. And there will always be natural mutants. So I think that's also a challenge that we have as researchers, that how do we interpret the natural mutants? In the times when we are doing genomic surveillance and all, we should be very careful how we are interpreting our data. So natural mutants will always be there, but they are not the cause of concern. The concern is when all the sensitive bacteria dies when they are exposed to antibiotics and the natural mutants get selected out. So in a forum where we are discussing a new antimicrobial, which has the danger of we losing it quickly, I think I would like to quickly take you through the innovation priorities for our own country so that we do not lose our drugs so quickly as is happening currently in our country and all other countries where we have over-the-counter availability of antimicrobials. And also we have an approach to health which is so different compared to the West. We have people who want to access healthcare right on day one when they have fever, which is not so in the West. So we are a very different country. We are used to real-time access to health. We do not wait for a week. Forget about antimicrobials. We don't even wait to see a doctor. So we are a very different country, and so our innovations in the realm of AMR have to be different. So we suspect AMR when we can see a microbe not responding to an antimicrobial, but it is always confirmed in laboratories. So it is very important to understand that in addition to new molecules, we have to invest in innovations in the lab space also. I'm sure some of you have seen I will quickly take you through the rest of the slides, but some of the important points I want to highlight here. In the lab space, we are still dependent a lot on technology, which is Western, which is European, and they are very expensive. Let me tell you, our diagnostics, we are heading towards POCs, but let me tell you, in the world anywhere, POCs are not there. If they were there, we would have been selling them in our country. Even the West is dependent on culture sensitivity for bacteria. And so why we are not able to do culture sensitivity across our country, because all the technology is, the automated technology is coming from Europe and it is expensive. So that's another area where we need to invest in. I'll quickly share with you in my that exact slide. And AMR occurs naturally over time, as I have said. However, the misuse of and overuse of antimicrobials is accelerating this process. So we have seen these figures. They come up in every AMR conference. These are the latest figures which have been published. 2019 data published in 2022. It says that AMR directly caused 1.27 million deaths globally in 2019 and contributed to an additional 4.95 billion deaths. So making AMR a bigger killer than HIV, AIDS, and malaria. Now, this data also is based on some statistical modeling, which capacity countries don't have. And the best part is that the United Nations General Assembly high-level meeting on AMR has decided on a target of 10% reduction of this level globally. Now, how can you make a global target happen if the countries don't focus on their own data and want to reduce it by 10%? So this is what we are focusing on now, and we are pushing these stalwarts who have made these publications to make the know-how of these statistical modelings which they use to create these kind of figures also to be available and build capacity within countries. By 2030, it said that AMR would force up to 24 million people into extreme poverty, and drug-resistant diseases could by 2050 cause damage to economy as catastrophic as the 2008-9 global financial crisis. So it is a crisis. And, a person who's sitting on the lab side will see it's a crisis. It is certainly a crisis. So the contributory factors in our country, believe me, it's not animal first priority. It is the clinical medicine. Every human being can go to the chemist and buy an antimicrobial. In the West, it's not so. It's not available. And hence, when I have students coming to my institution, I always tell them, the first question they ask me is, what are you doing for reducing antimicrobials in animals? I said, no, I need to first work on humans. So our use in India, clinical medicine is the highest contributor to AMR. Then comes the use of antibiotics as growth promoters in animals, veterinary medicine, agriculture, industrial practices, and now comes, of course, there is also this presumption that the antibiotics in the environment, which have been there right from the time we started manufacturing antimicrobials, they also lead to AMR. We still need to figure out which way is AMR flowing. It's moving from human sewage, or is it coming from the environment to the humans? So transmission dynamics is a challenge which we are now pushing in National Action Plan AMR 2.0 to study more deeply for our country. Inadequately regulated use of antibiotics. We have regulations, but we know it's our challenge. We still get antibiotics over the counter without prescriptions. And of course, substandard drugs with a lot of online sales, that is also a challenge. So it's a contributory factor, and the others are limited IPC, infection prevention control in healthcare facilities, poor water sanitation hygiene in the community, high use of antibiotics in hospitals, leading to emergence and transmission of resistant pathogens between patients in healthcare settings, and underdeveloped diagnostic stewardship. So this is another point I would like to share. We may have a laboratory, but it is not necessary because the clinician may not use it. So I've seen it in the best of hospitals, antibiotics are prescribed without sending samples of patients. So these are all areas which are leading to use of very broad spectrum antimicrobials, and that's how we lose them quickly. So this has been our journey in India. We started in 2010. We constituted National AMR Task Force, the Ministry of Health did, and 2011, the National Policy for AMR Containment was adopted. In 2013, AMR programs were started to understand what is the burden of AMR in India, not in terms of mortality, morbidity. That's a task which needs proper research. It's not easy to get mortality, morbidity data based on records of the patients. But this was primarily to see the common pathogens isolated from the clinical samples and what's the resistance pattern. 2016, we issued the treatment guidelines for infectious diseases in India. And of course, they are being updated now again. And 2017, through an interministerial consultation, National Action Plan on AMR was launched, which was aligned to the Global Action Plan. So these were the 6 strategies. As you can see, awareness, understanding for a country as big as ours, it's only awareness and education, which can really move something. And knowledge and evidence, we have enough evidence, and that's why in the National Action Plan 2.0, this has been changed to lab capacity building and infection prevention and control, optimizing use, innovation, and leadership. So I will just take you through in the next 2 slides what's the current status. So there are a lot of awareness campaigns happening across the country, but we need to do much more. We are a very big country. And I must say that this awareness and education has still not made a bent on the clinicians. What is the reason for that? Clinicians continue to write, and after COVID, it has become even worse. We're writing too many antibiotics on prescriptions, even for viral infections. So, one of the reasons is the lab. So, innovations in India can't be just on molecules, it has to be much more on laboratory diagnostics. Because, as I said in the beginning, the diagnostics in India, and we need not focus on point of care. Point of care is important, fine, but most important is making affordable diagnostic for our country, especially the blood culture systems. We are still dependent, and I must share with you, just a month or two back, one of my friends from Australia called me and said that they were dependent on a blood culture system which comes from Europe, and all the consumables in the whole country were not there. So you can imagine a whole country was waiting for consumables coming from another country, and they were not able to do blood culture systems. Fortunately, we in India still are on not just those automations, we still have a lot of manual methods, but we must create automated systems for blood culture in India, which is affordable, which is indigenous. We are really wanting to have diagnostics, even if it's routine culture. Point of care will take a lot of time, it's very challenging, and that's why it's not there globally. But most important is to get innovations, indigenous solutions for culture sensitivity in our country. So as I said, we have not been able to make a bent on the clinicians. That's because the laboratory is still not reached that level where the clinicians can really bank on them. And that's one reason why we are struggling, to have evidence-based use of antimicrobials. With this new molecule, we will have a challenge. The antimicrobial stewardship will be a challenge, which requires that the laboratories are used well and only based on, when you see a bug which is not responding to your other narrow spectrum, do you come to the recent bug, which should be used mostly for more severe infections. And then if you don't have a good lab, healthcare associated infection surveillance is also a challenge. And so I will give you a small example through this publication. This is a publication which happened it's there on PubMed, Changing Trends in Antimicrobial Susceptibility Pattern of Bloodstream Infections. Now this claims that this is the first such report from secondary hospitals. But I will share with you, on the right side, you see a map, which is the number of institutions we are working with from NCDC. And I must share with you, it's a very big challenge. We are trying to strengthen lab capacity. I have already shared with you the challenges. We do not have indigenous solutions. We are still waiting for expensive consumables and machines from abroad. And that is the reason the laboratory testing is not on mark. And we have a publication which comes from secondary institutions, which is very challenging. And we went ahead and found out that, sorry to say, that the report was the 8% vancomycin resistance, which was reported in Staph aureus, was wrong. From the whole country, we have found barely 3 isolates. And that also, we get many, but we try to confirm them. None of them gets confirmed. So one of the problems is the clinicians are not accepting what we are saying because of this reason. So we need to invest not just in equipment consumables, we need to invest in standards for quality, and we need to also have some way of validating international cutoffs for antimicrobial susceptibility testing in our country. Our pathogens are different, our PKPD will be different, our people are different. So we need to do some kind of validation. That should be another area of innovation. So for the third strategic priority was reducing incidence of infections, and a lot of capacity building happening. We have Kaikat program under the Swachh Bharat Abhiyan, where we go around looking at facilities, how is the infection prevention. And but however, what is missing is digitization. Healthcare, we have Indians doing digitization for healthcare abroad, but in India, we are still a long way to go to digitize our healthcare. And so we are not able to monitor. Institutions are struggling. Manuals, they still have manual methods, and the same is for capturing. So the fourth strategic priority is optimizing use of antimicrobials. Facility-based antimicrobial stewardship practices are being established in specific institutions, but we are struggling for surveillance of antimicrobial consumption and use in the country. This is because, again, we need digital solutions. So these are all areas of innovations in our country. So that if we don't know how much of antimicrobial is being used, how can I reduce? For that matter, when you bring this new molecule, I'm sure you would like to know where it's being used. We are not misusing it. And so digital solutions, both on laboratory diagnostics and on especially HMIS, that is Hospital Management Information Services, we should have it mandatory across the country, and we must have innovative solutions for that, which are easy to buy. There are states who have struggled. I know states have tried to invest, But they are so difficult. After 2 years, the person who has built it for them disappears, and then they don't know how to maintain it. So these are genuine problems, genuine solutions, which we are looking for. Sorry. Yeah. So these have been the implementation challenges of National Action Plan. We have had limited ownership from non-human health sectors. And one of the reason is that the National Action Plan 1.0 was every activity was attributed to every department and ministry, and that's why it was very difficult to monitor. And we had, for coordinating the entire National Action Plan, we were supposed to constitute a new body, National Authority for Containment of AMR, which was very challenging, which has not been constituted. You cannot bring a fifth silo to actually coordinate, to make them coordinate 4 silos. So this was not a very feasible option. And so we've removed all this in the National Action Plan 2.0. And there was no mechanism for sectoral, intersectoral coordination. All this has been taken care of in the National Action Plan 2.0. So these are my last slides. I would like to share. These are the WHO priorities, but let me tell you, they are more global. They don't focus so much on the challenges which we face in India. They're talking about rapid POCs, and I have already shared with you, point of care is something we should look at. Next level, first, we should look at the culture diagnostics, making them indigenous and affordable and accessible for us. Antimicrobial stewardship, they're talking about AMS interventions, burden, mortality, morbidity, impact on AMR. This is still a challenge. It needs very rigorous studies. And the quadripartite for One Health priority research on AMR has talked about transmission dynamics, which we are also talking about integrated surveillance of AMR, AMU, and behavior insights and change. So at national level, I would like to share with all of you and that we at the Public Health Institution and heading the program from the Ministry of Health would like to share, we need affordable automated blood culture systems with non-proprietary consumables. We don't need closed systems. They're very challenging. We need affordable automated, semi-automated bacterial ID and AST systems. Use of whole genome sequencing for AMR is another very big challenge. I see institutions like a White Elephant have gathered their Illumina machines, but then they basically have pipelines which are all international, no way to validate what they are seeing, what data is coming out. And so I personally don't push people. It should be only in the area of research that we work. We still have a lot of phenotypic work to do when it comes to AMR and digital solutions for real-time access of test results with clinical testing. Operational research, I would like to share morbidity, mortality are very difficult. And I said we need statistical modeling, which has been done in that publication in the Lancet to be brought to every country so that we all can do our own statistical modeling at the country level. And so that our figures match with what is given in the international publications. Impact of antimicrobials on normal flora. So first line defense is nobody's talking about. It is the first line defense, our flora, and that is a study which we really need. That has also been included as one of the priority in the National Action Plan 2.0. Impact of vaccines on immune system, very important to understand. I think as we read immunology as a science, we read that our immune system gets, when we are born as newborns, our immune system gets activated when they come across antigens. And there was always a balance between what do we vaccinate against and what do we let them develop natural immunity to. So that is also something we need to study deeply. AMR transmission dynamics and mitigation of impact of AMR in the environment is very important. It's a global topic, but we need to develop solutions for our own country. So this is my last slide where I would like to share with you that One Health approach is really one of the challenges if you want to contain AMR. So One Health approach has made one sector blame the other, but that's not what is One Health approach. And that's why the new word that has been put, integrated and unifying, is something that we from India are not very happy with. We still feel when the different sectors are at a different levels, we need more coordination rather than integration. You can integrate 2 sectors which are working at same level. If we had lab capacities in animal health and human health at par, we could have integrated, but it's not so. So I still would like to stick to the definition which was used previously, the collaborative effort of multiple disciplines working locally, nationally, and globally to attain optimal health for people, animals, and our environment. And it recognizes, we all recognize health of people is connected to the health of animals and environment. Thank you.
Thank you so much, ma'am, for presenting the strategic initiatives that will help India contain AMRs. I now invite Dr. Anand Kumar, the co-founder and CEO of Bugworks, an India, USA, Australia biotech startup working on tackling the problems posed by untreatable bacterial superbugs and being supported by global blue chip investors and AMR global grants to deliver his talk. Please, sir.
Good morning, everyone. I want to thank DBT Bairak, Dr. Jitendra and team for inviting me. This is an amazing honor for all of us from India to see an antibiotic, Nafithromycin, being launched by Wockhardt. Wockhardt is my super senior. We are a small startup company called Bugworks, sitting in C Camp in Bangalore, working on antibiotics. And we look at companies like Wockhardt, Orchid, and others for inspiration. And it's extremely amazing to see minister giving his time to launch an antibiotic. Nowhere else in the world are antibiotics launching except in India, because it's such a tough space elsewhere, and we have a great opportunity as a country to be world leaders in this space. It's just absolutely incredible. I also want to thank Bairak, because Bugworks started because of Bairak. And I see, Manish, Shilpi, Dr. Jitendra, Sanjay, these are folks I can never forget, because we wouldn't be standing here as a company had it not been for the seeding support from Bairak, right? So please give Bairak and Wockhardt a huge round of applause for what they did, right? So Dr. Lata has laid the foundation of what's going on in India and the challenges. The same challenges affords us opportunities that are incredible. As a startup company, I split my time between the US and India. And AMR in the rest of the world is a crash and burn. Most number of bankruptcies, most failures in biotech, lack of market, tough science, tough economics, that's AMR. Just switch this whole equation to India today, right? Almost 25% of the worldwide cases in AMR are India. So we have a huge problem. It's a hot spot for AMR. But that same hot spot has slowly becoming also a haven for solutions. Because I am of the firm opinion that AMR solutions that work in India will work in the rest of the world. Our pathogens are unique, our resistance patterns are very difficult, and when you do innovations sitting in Boston or Bay Area or San Diego or London without having stress tested it on our pathogens, it may not work when it comes here as a new antibiotic. So it's fantastic to see recent launches from India, right? N-metazobactam, Nafithromycin, the stories we know about Zainich that's coming up from Wockhardt. It's so heartening. It's so phenomenal. And this is going to change the landscape for the world. So why do I firmly believe that India is going to offer the solutions? This doesn't come from some nationalistic fervor, because that doesn't last. It comes from data. We have the patients here, which are the toughest to treat patients. We have clinical trial networks that are coming up to speed. We have to thank folks like GARDP from Geneva, who are working with ICMR, working with Bairak Biopharma Mission in trying to put together the best practices for clinical trial networks. Our regulators are waking up to the reality that we cannot be left behind as a country. Regulation has to move from a black box to at least a gray box before it becomes a stringent regulatory authority. We see accelerated pathways happening in antibiotics to where CDSO is saying if you have good, great phase 2 data, we may allow you early access into India. New thinking. Devil is always in the detail, but new thinking. 30% of the global antibiotics are produced from India. 60% of vaccine capacity is from India. You walk into a Walgreens or a CVS in the US, 1 in 3 chance that the antibiotic that's picked up there, generics, came from India. Manufacturing capabilities and capacities are in India. Most companies in AMR are failing in the world for 2 reasons. 2 reasons. One is lack of market, because you don't have the number of patients. Stewardship, obviously. And second is small companies cannot do CMC and manufacturing the way Indian pharma can, right? And Wockhardt, with that background of manufacturing, is able to bring that to bear as well. So our expertise in manufacturing and CMC, because cost of goods has to be affordable for the rest of the world. I cannot innovate for the West and hope that it's going to work for a 6 billion population that's living in low to medium income countries. So my request to Dr. Jitendra and to Bairak DBT is, we need a clarion call. All of the pieces are there, right? Innovation is happening, manufacturing, patient load is there. CDSU is trying to make changes in regulatory paradigm. if we lose this opportunity, I think we will regret it. This is the greatest opportunity for India to become a world leader, because solutions that are built here will work elsewhere. I'm being honest. Except for Orchid, Wokeheart, small companies like Bugworks, the rest of the world has very little new pipeline in anti-infectives, right? There is no pipeline. For 2,000 trials in oncology, there are 20 trials in AMR, right? It's incredible that there is no investment, no trial. So we have an opportunity to bring to bear things that nobody else can. And the multiplier effect on India by solving AMR is huge. AMR is not an infection problem alone. Most cancer patients in tertiary hospital die because they pick up an infection post chemotherapy or immunotherapy. We are a huge diabetic population. We are a huge immunocompromised population. The multiplicative effect of AMR is unbelievable. So if we are able to come together to solve it, and I'm very optimistic that we have the pieces together, right? And we have organizations like NCDC, ICMR working in very constrained environments, but trying to do unbelievable things. And as a small company, we are funded by the US government. BIRAC did the early funding, and we are funded by US government and now by GARD-P in Geneva. It's become very clear to the funders in the world that any solution in AMR has to start from India. It needs to be able to solve the resistance patterns, the clinical usage patterns, the limited diagnostics patterns which we have here. So this is going to become the solution hub. So Dr. Jitendra, my request to BIRAC as we celebrate this wonderful moment is that we need a clarion call. We need much deeper funding to pull a moonshot program for India, Dr. Jitendra, that says we don't need hundreds of millions of dollars. You know, we need probably a couple hundred crores, which is peanuts for a country that's fifth largest GDP in the world. A clarion call that DBT BIRAC can pull together that says we are going to invest in diagnostics, discovery, stewardship. We already have the pieces. We just need a little bit of support. Bring CDSCO to the table so the regulator is there along with us, so that stewardship is also made important. Maybe we need a Schedule H plus in India, so that new drugs don't get destroyed in 2 years because we use it and abuse it over the counter, right? So a clarion call to do a moonshot program. If genomics is so great the way it is, if oncology research is so way the greatest, is because countries called it a moonshot and said we are going to solve this problem. Today, if India doesn't do it, when can we do it? We have companies, we have great talent pool, we have the patient load, we have clinicians who are used to working in the toughest conditions to support us to work to take innovation forward. So the pieces of the puzzle are there. This is not a blue sky research. It just needs a little bit of hand holding and support. If we pull it together, absolutely no doubt that India will lead the world in AMR innovation. For God's sake, we launched a hypersonic flight day before yesterday. We're launching the best satellites in the world. We're doing great work in IT. Yet, we are saddled and riddled with AMR. And wherever I go in the world, they tell me, a country like India that does so many great things, why are you guys the worst AMR problem area? We are going to change this statement on its head. We're going to become the solution providers, not just known as the worst AMR hotspot. Together we can do it. Congratulations to Wokeheart, to BIRAC, and we march ahead. Thank you.
Thank you so much, Dr. Anand. It is reassuring that we are in the right space and time to address issues not just for our own population, but the world at large. So with this, I now call upon Dr. Gopal Nath, Professor of Microbiology and Dean of Research at the Institute of Medical Sciences, Banaras Hindu University, and one of the pioneers in the field of bacteriophage therapy, to deliver his talk.
Very good morning to all of you. It was really a great pleasure to me, and I would cannot stop myself telling that it is 90% times I got the funding from DBT itself. This time also, whatever we are doing is being recognized by the DBT BIRAC. So being a medical microbiologist, I think 90% of the funding I got from here. And then I'm sure that you must be knowing something about the bacteriophages. Or should I give some brief about the bacteriophages, what they are? So it started from the river Ganges and Yamuna, you know, by the Henkin. And then what we did is that everybody feels that the Gomutra is very sacred. Then I started working on the river Ganges. Then I found that the bacteriophages richest source is the sewer water of the hospital. Then I came to just look upon the Gomutra, what is there special. So I tried to isolate the bacteriophages against the escape organisms, and I found that they are rich. And then it was not, the science says that it's not the cow only, why not to buffalo, why not the goat? So I screened the 5 different goats and buffaloes. I found the same bacteriophages were there also, which is present in the cow. And then I screened all the 18 members of my lab who were working with me, their saliva, their urine, and the bacteriophages. We are having rich bacteriophages in our body. So before this, if I say about the microbiology, the total number of eukaryotic cells in our body is 10 power 13. The bacteria are 10 times more than the eukaryotic cells, and the viruses are 10 times even more than that. So basically 99% of, by number wise, our body composition is bacteria and viruses, and only 1% is the human cells. So this is the composition. And since beginning, as previous speaker told, that the immunology starts basically just after birth against these microorganisms. So when you are having the pollination at the early 2 years of life, you are not going to, the antigens are those immune cells are getting tamed. So they are not reacting to our own indigenous flora. So this is the story, and the viruses are there, and the antibiotics and the bacteria are basically, you see, this is the bacteriophage. So they are going and they are causing 2 types of the cycle. One is the lytic cycle, and another is the lysogenic cycle. And then we are utilizing the advantage of the lytogenic cycle, the lytic cycle, because they are killing the bacteria. So there are 2 systems present in the nature. One is the antibiotic molecules, another is the bacteriophages. So we, if define them, antibiotics are basically signal molecule which basically gives the signals in the stressful condition at sub-inhibitory doses. The human being, we just increase the concentration of those molecules and killing the bacteria. But bacteria evolved the system to resist them. Similarly, the bacteriophages present in the nature, 40% of the all bacterial cells are eliminated every day by bacteriophages, because they are an essential component of our food chain. That is essentially needed, that's why they are doing. And then they are basically the genetic engineers in the bacteria. They are producing a lot of variants, and in different stressful conditions, at least 1 or 2 or more variants are getting survived out of those stressful conditions. So basically they are for the bacteria, and they are working in close association and collaboration. We are taking the advantage just to increase the number when needed if some bacteria is causing problem to us. So these are the 2 philosophy. One is the signal molecule, another is the genetic engineers of the bacteria. They are the bacteriophages. So these are the different classes of the bacteria which I used to teach. The mnemonics are there where they are working. I'm not going to detail of this one. I shall be rushing because, and these are the different sources of the antibiotic molecules. And this is the story how we developed the antibiotic molecules from 1940 to 1980. Now very few new molecules are there in pipeline. And then we are just trying to find out the alternatives. And then in 1966, one of the surgeon, because they are not having microscopic vision, so they said that the time is over for the this AMR, like the bugs, and that we are going to treat them because we are having penicillin with us or some more molecules. But that will never happen. The fight will continue. The war will continue with the human being or the any eukaryotic cells and the bacterial infection. So man is always wrong. We think that we are the most genius individual, means creatures on this earth, but that is never happening. So there are superbugs, and then this boy is thinking that every problem has, and every dot has got its antidote. So there are many antidotes. We know that the peptides are there, bacteriophages are there. So this is the story of the bacteriophages. And very simple it is to isolate. Any lab can do it. Just make a lawn culture like you do in the antibiotic sensitivity testing. Put the sewer water or Ganges water or whatever you find, the dirty water, what we say dirty means number of microbes are more there, that is the dirty water. So we can get the plaques maybe on next day or day after that. So these are the viruses which isolated. We got lysogenic, lytic, and then if you see, these are the plaques. If you increase the number, you are getting more plaques. If you increase further, clearing up the plates. So I am reporting here a case. Luckily, he is the scientist in DST. I was thinking that he is in DBT and working in Delhi itself. His mother got sick and she was going into septicemia. And I shall be rushing again this because after the orthopedic implant, she was having some biofilm formation. And you see that what happens to that case. So she got injury 12 years ago, then again another injury, then cyst formation was there. And then the scientist working, wrongly I have written DBT, but he is in DST, Government of India, and he was knowing somehow about my work. He contacted me, and by that time, I was having a PhD student submitting his thesis. 3 well-characterized whole genome sequence bacteriophages were there with us, and purified also. So immediately we supplied empirically. And you see, this is the level of the procalcitonin. This is the septicemia marker. And if it is more than 10, it is really very dangerous situation, but in between, it is really serious. So what happened? Her procalcitonin was 5.25, and then you can see the blood urea and the serum creatinine level that was showing that the kidney was also getting compromised, and she was going into septicemia. And we started the therapy, and then empirically we gave, later on we tested, and all the 3 phases were working. And we gave like this to the wound side. I shall be showing you that one also. So on day 5, you can see that the total cell count from 28,100 on 2nd December came down to 15,600. It was just reduced in 3-4 days only, and procalcitonin also came down to less than 1. And the serum urea and the creatinine also became normal. But we continued this therapy. This was the wound, and having a cyst infected, and the Klebsiella pneumoniae was resistant to all the antibiotic available. So he was really scared, and he was thinking that he is not going to save his mother. That's why I told that you give the consent from the true treating physician, and I can give only on the compassionate ground. So that was started. That is the story I'm telling you. Because only 10 minutes is there, so this is why I'm rushing. And then this we kept on giving. You can see the different levels coming down. This is again when the GC, general condition, improved. Then the surgeons decided to open the wound, and then they debride it, and we started giving, kept on giving the bacteriophages daily with the same dose. And then now you can see there is fluctuation in total leukocyte count sometimes, but there were certain regions because in between there was infection by Acinetobacter baumannii also, and once we detected the UTI also with the Enterococcus faecium. So whenever there was this incidence, the rise on the TLC was there, but the procalcitonin and the blood urea and serum creatinine was normal. So I shall be just showing you the other cases. Then when everything became normal, the total count and every this inflammatory cytokine markers came down. So surgeons decided to close down the wound site, and then they put a catheter to push the bacteriophages inside, and we kept on giving daily doses. This was the, and bacteria kept on growing, but at with very low number, scarce number. This was the situation there. And this was the tubing by which we are pushing, and we are there was daily serous discharge about 100 to 200 ml. This was huge wound actually. So this was like this, and you can see how the serous fluid was coming out. This was the speed. This was on day 20, and then we were culturing every day. So this is the wound. This was the collection. On day 29, this is the wound, and this is the total leukocyte count. You can see that this is within normal limit, 4,000 to 11,000. But CRP was still higher, and there was growth of Acinetobacter baumannii. So we again having the another PhD thesis, having the well-characterized phases with us. So we started giving that one also testing, like antibiotic sensitivity testing. So she was lucky also. And then the Klebsiella pneumoniae kept on growing, and then we kept the therapy continued for 152 days until no growth was observed in 7 consecutive samples collected from this. This was the end of this one. So this was almost 5 months it took to eradicate, because there was biofilm formation, and biofilm cannot be eradicated by the antibiotic. Cannot be eradicated. I am using very forcefully that antibiotic can't do this. It is the bacteriophage only which can do it. So we stopped this fast therapy, and then we did. This is the end of the therapy, and she was cured, and then now she is moving like this. She could be saved. So this was the total journey. You can see, whenever there was another infection or UTI, there was a rise in TLC, and then it came down to the flat situation. This is the different conditions of different markers you can see, and procalcitonin and the this blood urea, serum creatinine came. Yeah. So this is another case after taking consent, local wound infection. This is from Hyderabad. He is a father of one of the medico, and then he contacted me because he came to know that we are doing this. This is staph aureus infection. We produce, means because there was a long time with us. So we isolated the specific bacteriophage, purified it, and then started therapy. You can see the result. You can see the osteomyelitis was also there. Bone chip is still there. That was cured. So this is the end result. Otherwise, the surgeons were suggesting for amputation. So we have treated like this more than 300 cases till date. There were 3 PhD theses have been submitted on the local application chronic wound, which are associated with the biofilm formation. So you can see there are many more cases I can show you. This is the randomized case control study, because I talked to the DG ICMRD, then Dr. Balram Bhargava. He said that you keep on collecting more and more data, then we'll decide the regulatory body can do something. So Dr. Anand told that the CDCSO must think about this also, because they are the part of our body, and it's not that we are going to follow the US only. We, the Indians, can decide our own policy for this. So when we'll be doing this, I don't know. We are just simply workers. We are the labor class people. The policy decision makers are different people. So you can see when it started the therapy, it also, yeah. So I'm just, there are many more publications, but one more thing before finishing my talk is that very encouraging that when you are using the bacteriophage and antibiotic for synergy, it is really fantastic. We should not leave the hope, and this is a very hopeful condition. Even colistin, if you are using at the sub-inhibitory doses, but the bacteriophages first. And after use of the bacteriophages, you are using 6 to 8 hours after these antibiotics, which are not really responding. Their MIC has gone much higher. Even at the lower MIC, they are able to eradicate. We did a lot of in vitro studies and in vivo studies. Time is not there. Otherwise, I could have shown you the wound infection and the UTI infection, where you use the bacteriophage first, and then 8 hours later, you use the sub-inhibitory dose of the antibiotics. It is really magic, fantastic results you can get. So I'm so hopeful about this, that AMR is not going to kill us in future, provided that we are accepting the bacteriophages also. You can see this other. So this is the end of my talk. Thank you very much.
Thank you, sir, for sharing your insights from the clinical trials on wound healing in the critical cases. We hope to see such alternate therapeutics in the practice soon. So thank you so much, sir. Our next speaker is Dr. Bhabatosh Das, Professor, Translational Health Science and Technology Institute, Faridabad. A fellow of the National Academy of Sciences in India, Dr. Bhabatosh is a recipient of many prestigious awards, including the National Bioscience Award and the BMJF Awards. My only request to him is to please stick to time, because... please.
Sure, Dr. Neha. I'll try to finish, I hope, by 10 to 11 minutes. So thanks, DBT and also the BIRAC for this invitation and also giving me this opportunity. So Already all the speaker explained that how actually AMR is creating issue not only to the health sectors, but if you look to the AMR, so it's an equal problem to the other sectors. Other than infection, particularly the organ transplantation surgery, that is one of the major issue, food industry and livestock. And there are sets of pathogens you could find that those are the common, but you do also find there is a distinct pathogens, those are linked with specific types of the problems. And when we are talking the AMR, this is one important information actually we all need to understand, that what are the functions that contribute to the antimicrobial resistance. It's a very complex phenotype. It's not like that only one proteins or function can contribute. For the same, antibiotics could be neutralized with the multiple function. So if we really need to tackle the antibiotic resistance, we need to first understand the antibiotic resistance encoding function into the genome. So it's my lab in Faridabad, we are mostly investigating on the genomics and metagenomics on last almost like 10 years. So what we are trying to do that if we could exactly identify what are the functions or pathogen specific functions that contribute to the antibiotic resistance, we can develop a kits for diagnostics, but at the same times we can also develop some in silico compound that can inhibit the resistance functions and it could act as an antibiotic potentiator. So today in my talk I'm just mostly going to discuss about the antibiotic potentiator, so molecule that can resensitize a resistance bug and the existing antibiotic can work. So we started this antibiotic resistance work, and what initially we did to have an holistic idea what types of resistance pathogens are circulating in India, both in the clinical settings as well as environment. We have collected more than 6,500 bacteria, those are human associated. This is almost covering more than 120 different bacterial species, and these are from the human isolates and the environmental isolates. Each and every isolate first we confirm them by 16S RNA sequencing, and then we selected pathogens, those are specific to the humans, those are common both human and environment, or also to the environmental isolate for the genome sequencing. Until now almost like 2,500 complete genome sequencing been analyzed and also been published. So when we did this analysis part, what we observed that you have the resistant genes, those are very pathogen specific, although the resistance phenotypes are common. Just for example, if you look to the beta-lactam resistance, if you are looking at Acinetobacter baumannii or Klebsiella pneumoniae, both of them will give you the phenotype of the beta-lactam resistance. But question is that whether the same functions present in both the bugs or is there a bug specific resistance function. So what we observed that there are certain functions, those are very bug specific, like if you look to the Acinetobacter baumannii or Pseudomonas blapav is the very common, but if you look to the Klebsiella pneumoniae, oxa is the common. Phenotype is the same, but the function is the different. So that actually make a major challenging for the molecular diagnostics. You can do phenotypic testing, but unless until you have a very conjugate probes, most possibly you will miss them. So the same phenotypes has also been reported from the other parts of the globe. If you look that when actually in the study they analyze the Klebsiella pneumoniae, Acinetobacter baumannii and E. coli, what they do observe that there are certain resistance functions, particularly if you look to the particularly that confirm the resistance against beta-lactam antibiotics, these are very common. But if you go for the ammonium acid resistance, there are certain functions including AD5s, AD2, those are very pathogen specific. So all the cases, so this is not only the India, wherever you are going, whether it's from the India, Asia and Africa, and we have also included close to 3,000 isolate across the globes, and we find the same things. So what we are trying to do in our labs, that how to actually neutralize those resistance functions. If you have already resistance bug and if the existing antibiotic is not working, is there any way? So the bacteria is the not problem. Problem is the resistance function. And most of the cases, if you look to the escape pathogen, the resistance are happening because of the acquisition of specific functions. Those are enzymatic in nature. So if you could simply inhibit those enzymes that confer the resistance, the existing antibiotic will work. And the best practice or the best example is the beta-lactam inhibitor. Those are available into the market. So these bacteria generally don't respond to the beta-lactam, but once you add the beta-lactam inhibitor, the same beta-lactam antibiotic work. So we are working on that aspect. So other than beta-lactam at the clinics now, there is no potentiator. So we are looking for all these different antibiotic classes and developing the antibiotic potentiators. So when you want to develop a potentiator, the first and most important things that you need to have a very robust and sensitive assay. Your assay in such a way you need to design that it should take minimal times, otherwise your compound may not sustain. So what we did that we have initially treated with the E. coli, but we identified other bacteria that grow faster than E. coli, so we have selected the Vibrio cholerae. Then not only the resistance function, there are certain functions that present in the bacteria, those cause the intrinsic resistance. So particularly if you look to the small molecule PPGP and efflux pump, so they pump out your molecule and that you lead with the false classification. So what we did, we actually deleted all the functions that cause the intrinsic resistance of the antibiotic and make the cell very permeable for all the antibiotic. So antibiotic can easily go, at the same time the antibiotic potentiator also can reach to the target and get their function done. And the fourth point that we also looked at the assay sensitivity and the detections of your assay. So we have both the chromogenic assay as well as the turbidity assay that can detect that whether your compound is working or not. Okay, and then finally we are going for the reproducibility of your assay. So till now, almost like 27 antibiotic resistant genes that we manipulated in the genome of a very sensitive bacteria, particularly in the Vibrio cholerae, and then we screen. So normally when we are giving this resistance function and the antibiotic, there is no effect in the microbial growth. But if you are giving a compound that normally not antibiotic and in combination with antibiotic, they inhibit the growth, that means they actually inhibiting the resistance functions. So we have screened around 4,500 compounds. Those are FDA approved compounds used for the other purposes. And ultimately we identified around 18 compounds. Those have these potentiation activity and they can reduce at least 80% of the activity of the AMR. And currently we are focusing on one of the antibiotic resistant potentiator, that is the against beta-lactam BMM, that one of my PhD student Dr. Minal is working. So here what we observe that these compounds normally it's not the antibiotic, but if you are putting with the your spectromycin, they actually resection the bacterium and the spectromycin work. We have tested with both the reporter strain E. coli as well as the Vibrio cholerae, and both the cases we have the similar functions. We have also done the dose optimization, and what we observe that as minimal as like even if you are adding 2.5 micromolar concentration, it completely inhibits the growth. But if you are giving only antibiotic or if you are giving only potentiator, there is no effect in the growth. We have tested then with the different types of extensive drug resistant clinical isolate, including E. coli, then Klebsiella pneumoniae, Acinetobacter baumannii, and Pseudomonas aeruginosa. And all the clinical strain what we observed, the BMM3, that what we identified, they have the antibiotic protection activity, and they actually potentiate the activity of spectromycin, gentamicin, and erythromycin. So in summary, what I can say that now we have a very sensitive reporter strains. This is a high throughput strain that you can use for screening the compound. And even when you are actually developing a new antibiotic, and if you really want to know how many resistance genes they cannot neutralize, you can use that systems. With that, I would like to acknowledge all my collaborators and particularly to the DBT for generous funding. And I'd also like to thank my lab member for this work, and thank you for your attention.
Thank you so much, Dr. Bhabatosh. Without further ado, I'd like to call our next speaker, Dr. Aradhana Vipra, Head of Microbiology, Gangajan Biotechnologies Private Limited, for her talk, please. You're going to change the slides for me?
Yeah, so good morning, everyone, and I just would like to thank Dr. Jitendra Kumar for having me here at this historic event. So in next, I know the time is constrained, so in next 10 minutes, I'm going to just take you through about what Gangajan is all about and what we are doing, our different phage technology platform in the space of animal and plant health. So Gangajan is founded in right way back in 2000 by Professor Ramchandran. He was a visionary by himself because he's the first person to bring in phages, bacteriophages in India, and then we started working on bacteriophages since then. We have a team of around 30 to 40 scientists coming from a different backgrounds of biotechnology, biological sciences, which is typically a setup that we need for any drug development or discovery kind of a work. We also have our own animal house where we do lots of infection model to test our drugs. We also have been, having lots of intellectual property asset that is a typical asset for any biotech company per se. So basically, the lab is the R&D is located in Bangalore. We have our parent company based out of Singapore. So during this whole period of 20 plus years, we have collected different number of bacteriophages. So I'm not going to tell you all about what bacteriophages are. I think Professor Gopalnath has given a very good background on the bacteriophages per se. So we have around more than 500 different bacteriophages for different gram-positive and gram-negative bacteria that we have collected across these 20 years. And I think Gangajan is one of the biggest repository of bacteriophages per se. We also transfer these phages to different companies under MTA for in vitro testing only. Basically, we started with phages, but now looking at the different regulatory aspects of phages being coming for a human therapy, although there is a lot of talk about bacteriophages today, but 20 years back, this was not the case. So we started working on phage-derived lysines. We called it lysines. So we have taken first of that kind all the way in human to 2B, phase 2B clinical trial. This was for eradication of MRSA. We've also been recently granted funding. We've got a funding from Carbex where we're working on another type of antibacterials called bacteriocins. These are for Klebsiella pneumoniae, which are involved in hospital-associated pneumonia. So this is a part of the phage-derived lysines or bacteriocins. In terms of bacteriophages, we are working on multiple platforms right now. So we are looking for bacteriophages basically in the non-clinical use, where we are developing the phage platforms for poultry, for salmonella infections. We are also looking for different infections in aquaculture, also in the plant bacterial infection, and also to treat environmental sewage samples. We are also looking for bacteriophages in the home care hygiene space. We are looking for bacteriophages for malodor causing bacteria. So these are multiple collaborative kind of projects where Gangajan offer expertise, and we've been working with our collaborators to develop these kind of products. So I think just skip this slide because we know AMR is a huge problem, and there is a whole lot of talk about the One Health, which is also a clear mandate from WHO. So we also need to look into the different health system other than human health. It's also the environment and the animal health per se. So if you look at the antibiotic use in the animal husbandry, they have been used in multiple different ways. But the most critical factor that causes AMR in the animal husbandry is the way the antibiotics has been used as either the prophylactic or the growth promoters, where they've used at very inhibitory concentration, and that's what causes the AMR issues. So in terms of how we approach to each of these solutions, we work a lot on the lab way, where we isolate the different type of phages, we characterize them. We have also done the scale-up optimization for our salmonella phage product, and we've been working with other phage product in the same way. The most important part is to go and talk to the stakeholder, because these are the people where we are going to introduce our product. So that's the most critical step, as to know where your product is going to be applied and what is the exact need of the stakeholder. So just quickly talking about the technology platform, this is the first one that we have developed for salmonella in poultry, where we have went all the way for the scale-up optimization. We have also worked on the propagating host, and we have done some field trials in the real poultry case, type of setup, where we have shown the efficacy of bacteriophages in terms of different the growth parameters that the typical poultry farms look into. We've also worked on the vibriosis aspect in the shrimp aquaculture, where Vibrio is a major causative organism, and we have shown a good amount of data to show the phage efficacy. I'm not going to really go into details of each of them. We've also worked with Oil India Limited. They have a issue of this corrosion of their pipelines, which is caused by an anaerobic bacteria called sulfate reducing bacteria, So we have worked for isolating the phages for these anaerobic bacteria, and we have shown a small proof of concept of a study where you can see in this diagram, this black bottle is all about, having the water contaminated with sulfate reducing bacteria, and when we treat with bacteriophages, it really gives as good efficacy as the typical biocides that have been used in the industry. We're also working with the collaborator in the FMCG space for treating the malodor causing bacteria, which are also a critical important parameter in any case, any way of home care and hygiene. So there we are, this is under development where we are developing bacteriophages as a sustainable solution to treat these malodor causing bacteria. So there are lots of challenges when you are developing such kind of non-traditional antibacterials, as we call it as. I've been working with phages for past 21 years, and we have seen so many ups and downs during this whole journey, and the way the bacteriophages have been spoken about recently, and today is the best example that we are talking about bacteriophages from such kind of platforms. It gives me a lot of positive thoughts that this is going to be also another important antibacterials that we would be looking for. What I would like to emphasize upon is the regulatory type of a concern, because the product that we have been developing for plant bacterial infection, we have been talking to the regulatory authorities for the same, and what we understand is we really need to spread this awareness of the bacteriophages being looked into the alternatives, because I know if you go to the human kind of a phage therapy, there has to be a lot of study that need to be still carried out, because a lot of typical parameters of pharmacodynamics, pharmacokinetics and all those kind of things need to be really well studied. But in terms of environmental applications, as I would say, so, the plants or the animal husbandry, this is going to be a very good platform that we need to really, the regulators need to be really aware of and could start thinking about this. What I also understood is there are different parameters which you really need to work upon in discovery and development of these kind of non-traditional things. Basically, these are all live entities. So you need to really work around the formulation part, because you really need to make them stable. The scale-up optimization is also because these are all biological systems. So you are working with live bacteria, you're working with live bacteriophages. So, these kind of work needs lot of standardization in scale up. You need to put in lot of efforts to that. Also the product application, because the end point, the end user, they just don't want to, disturb their typical protocol. We have seen this, especially in our poultry kind of application, when we went and spoke with lot of these stakeholders. They weren't aware of what bacteriophages and what are these new antibacterials, and also they just don't want to change their typical protocol. So these are some of the things which we need to really keep in mind. And also the margins, the profit margins are very narrow. So you need to really work around when you are developing such kind of product to think about these kind of issues also. So with that, I think I will conclude my talk. Thank you so much.
Thank you so much. We really hope these alternate therapeutics come into practice soon, and we're able to have some differences. So our last speaker for the day, for this session, is Professor Urmi Bajpai, Department of Biomedical Sciences, ANDC College, University of Delhi. Her research interests include the study of bacteriophages and the encoded lytic enzymes as adjuncts or alternatives to antibiotics.
Thank you thank you the organizers for having me here, and a great opportunity and platform. The advantage of speaking in the end is that most of the good content has already been spoken, so that makes my life easy. And also that you've been saturated of hearing a lot, so I'll try to keep it short, but I have to introduce something which perhaps has not been talked today as yet. So the topic I'm going to discuss today, as bacteriophages have been very well covered, it's from lab to lives is the session, but I'm taking you back to lab because I'm not yet reached neither to the lives or to industry, but we are in academia and we are doing the primary discovery based science. And when we look at the antibiotic era, I just want to tell that in case, as Dr. Gopalnath is sure that antibiotics are going to work, even if they don't work, phages are there. But we certainly don't want to reach an era where molds are being used to treat infections. So we know that why this date is so important or this week is so important. Challenges of present time, we know that because of animal farming and so many other urbanization, deforestation, the conflicts, war zones, etc., there is a spread of AMR genes in the environment, and that's where how there is an alarming situation. And One Health approach has already been talked about by everybody, and so we have to look at this, that not just human medicine, but how animal and human medicines and the resistance pattern impact each other. So this is very good, and as Dr. The first speaker spoke, that we need to have more of a coordinated approach than integrated, but I'm going by the definition. So the solutions which I want to focus on is populating antibacterial pipeline with non-traditional solutions, which is, I think, the theme of the session. And as Dr. Anand Kumar rightly said, that we have everything with us. We need to just we have to connect the dots and put the expertise together. So being a teacher, I'll take you to the classroom to tell what bacteriophages are. As the name suggests, the phages are those viruses. They are good viruses, like enemy of your enemy is your friend. So the viruses kill bacteria, and because they can kill bad bacteria, that's why we are interested in them. They are very abundant. So unlike the antibiotics, which are few in number, we have almost an inexhaustible supply of phages in nature. So we can always go back to them. I work on mycobacteriophages, and we got a fillip in 2019 when there was a first case in the whole world where a human patient was treated with engineered mycobacteriophages, and with very good results, very good success, and that's what jolted people to to put more impetus on mycobacteriophages. So we discover from dirty spots, as Dr. Gopalath has already said. Over a period of time, we have developed a bank of about 350 mycobacteriophages, which are not all genomically characterized, but with crowdsourcing interns and PhD scholars, we are developing this bank. And we believe these phages can be used for any sort of application. After the characterization, especially after genomic characterization and annotation, we are interested in those enzymes in phages which can cause lysis of bacteria. So the bactericidal proteins, we call them endolysins. So we are interested in several proteins from mycobacteriophages, but we are focusing for this talk on endolysins, which are, as Dr. Aradhana also mentioned. So just to take you through this slide, where you can see that the structure of mycobacterium is very complex. The cell wall is very complex, and hence bacteriophages make two kinds of enzymes to break open the wall of bacteria so that they can get out and infect more. So how about we recruit those enzymes as antibiotics? So enzymes as antibiotics, the term is antibiotics or protein antibiotics. And these are gaining traction because of several advantages they offer. One, there is no known resistance, though it's a very new technology, about 20 years old, only 20 years old, so it's still in infancy. But so far, no resistance has been reported against license, and that is the major USP of license. Of course, they can kill both antibiotic susceptible and MDR pathogens alike. They can inhibit and disrupt biofilm, which is again a very strong positive factor about them. And they have very rapid action and very high precision. So they don't cause collateral damage like broad spectrum antibiotics. Also, they are very safe, and because of which they are safe for commensals, and they also synergize very well with the antibiotics. And they have definite pharmacokinetics, unlike phages, which have auto-dosing property. So if we look at the structure of protein, because we are interested in them, so we need to know more about them. They can be both multiple domain or single domain. And the domain architecture, when we study, so this slide shows that in nature, we have huge diversity of these endolysins. And by looking at these colored boxes, you can see these colored boxes as domains. So these shuffling of domains can create very many chimeric lysins, and hence I believe that endolysins are present in nature in abundance, plus the chimerics that can be formed from these can give you an enormous broad base to screen and find lead molecules. The targets in peptidoglycans are those targets which usually bacteria will not evolve to change. And then these are the assays we do with the recombinant proteins, which are, they're chromogenic, they are microtiter based, they are high throughput and manageable. So after screening them, we identify those which are good, show good in vitro activity. Though we find that in vitro activity and antibacterial activity is not necessarily correlating. I mean, those which give fantastic in vitro may not give antibacterial, but we check their planktonic activity, we check their activity against biofilms, both their inhibitory potential as well as disruptive. And we have been able to identify 2 lead molecules which are able to, but though their concentration is still a little high, to 1 to 2 micromolar, but their biofilm effect is very, very strong. And so therefore, we also, so when we look at the synergy of the endolysin, so we find that when, let's say, we are treating endolysin B with rifampicin, and we are able to see the MIC coming down when we use them in synergy. So that's where we feel that not only the endolysins themselves can be very effective protein antibiotics, but they can also bring down MIC, and they can also perhaps bring down the concentration of the drugs which are very toxic and which are to be taken for a long period of time. This is another example where you can see that the MIC shifts because of the combination of lysine B with the rifampicin in this particular case. So endolysins are fantastic. Of course, there are challenges. The molecules are great, but we need to engineer them and formulate them so that they can be used in therapy. And their formulation and targeted delivery is one of the key challenge which I find. Potential immune response is often asked, but it's not been reported yet. And their large scale production and associate costs and alternate expression platform is something we need to work upon, because if we can find great expression platforms, then the cost can also be brought down, the safety profile of the proteins can also be can be taken up, and they can be more cost effective. And as Dr. Aradhana mentioned, that regulatory framework is the key, but then as you know that there are protein antibiotics or protein biotherapeutics already in market, so that's a relatively safer route than bacteriophages. And that's why I feel that, if we can work on endolysins, then they have several advantages even against phages, because they don't have auto dosing, and therefore their pharmacokinetics can be more definite. And with that, now this is a busy slide, but just to give you an idea that in the recent years, several startup companies, a lot of money is being pumped in these companies to develop these endolysins as therapeutics, not just for humans, but for animals also. And this is a case study by a Lysando, where animal was treated with endolysin. And in I want to mention that in Ukraine, there is a nasal spray being used with the license spray is being used for treating MRSA in the Ukraine soldiers, Ukraine war. So we are finding their application not just in medicine, but also as food pathogen control agents. And so, as I said, that I'm very glad to get this platform, because still we mostly invest in AMR on diagnostics, very important, small molecules, very important. But there's a lot of biologics which are present in nature in abundance, but we need to create awareness about those. And that's why we keep holding these talks and webinars and open lectures to tell people that how important phages are if we want to get some alternatives in quicker relatively time. And so with that, I would like to end my talk by thanking all the collaborators, my funding agencies, and this lab where we have PhD scholars and undergrads both contribute to this research. So one word I want to leave with is that phages and endolysins have a lot to offer. What we need is bringing people together and leveraging each other's expertise, because I think all of it exists in the country, but that interest and a scaffold is needed which can bring together different experts and perhaps develop into a technology in the coming decade. And maybe it can be a very good fight to AMR. Thank you very much.
Thank you so much, ma'am. So with this, we now come to end of the first session. As a token of our gratitude, we'd like to present a memento to our esteemed speakers. May I request MD Bairak to please present a memento to our first speaker, Dr. Lata Kapoor. Can we have a big round of applause, please? Thank you, sir. I now request MD Bairak, sir, to please present a token of our gratitude to Dr. Bhabatosh Das. Thank you, sir. May I now request you to please present a token to Dr. Anand Kumar, please? Thank you, sir. I now request you to please present a token of our gratitude to Dr. Gopal Nath. May I request you to please present a token of our gratitude to Dr. Archana? Thank you, sir. I now request you to please present a memento to Dr. Urmi Bajpai. Thank you so much, sir. We could perhaps have a group photograph before we proceed further. Can I request Secretary, sir, to please join the team on the dais, please? Sir, can you please join here for a group photograph? Thank you so much, sir. Thank you so much, everyone. So before we move on to our next session, which is the inauguration of the antibiotic, I request the people sitting at the back to please come forward, some of you, so that the hall is a little better full. The people in the last line could perhaps just take some of the seats in the front. And I request everyone to please be seated. We expect to have the chief guest for the day with us shortly. Good afternoon, everyone. So I request Honorable Minister Sir, Dr. Rajesh S. Gokhale, Secretary DBT, Dr. Jitendra Kumar, MD Bairak, Dr. Habil Khorakiwala, Founder Chairman Vocard Group, Professor Y.K. Gupta, former professor at AIMS and presently the President of AIMS Jammu, to please join us on the dais. On behalf of the Department of Biotechnology and BIRAC, I cordially welcome our Chief Guest, Honorable Minister of State, Independent Charge, Science and Technology, Minister of State, Prime Minister's Office, Personal, Public Grievances and Pensions, Department of Atomic Energy and Department of Space, Dr. Jitendra Singh Ji, in this program marking the World AMR Awareness Week. May I request Dr. Anamika Gambhir, Advisor, Department of Biotechnology, to please facilitate our Honorable Minister with a potted plant. I once again request Dr. Anamika Gambhir to present a welcome token to Professor Y.K. Gupta. Thank you, ma'am. May I request Dr. Jitendra Kumar, MD Bairak, to present a welcome token to Dr. Habil Khorakiwala. Thank you, sir. I request MD Bairak, Dr. Jitendra Kumar, to present Secretary DBT, DG Brick, with a welcome token, please. I once again request Dr. Anamika Gambhir, Advisor, Department of Biotechnology, to welcome MD Bairak with a welcome token. Thank you, ma'am. May I now call upon Dr. Anamika Gambhir, Advisor, Department of Biotechnology, to kindly deliver the welcome address.
Honorable Union Minister of State, Independent Charge, Ministry of Science and Technology, and Ministry of Earth Sciences, Minister of State, Prime Minister's Office, Ministry of Personal and Public Grievances and Pensions, Department of Atomic Energy, and Department of Space, Shri Jitendra Singh Ji. Respected Secretary, Sir, Dr. Y.K. Gupta, former Professor and Head Pharmacology, AIMS, presently President of AIMS Jammu, Dr. Habil Khorakiwala, Founder and Chairman Vocard Group, Dr. Jitendra Singh, Managing Director BIRAC. We have the distinguished presence of Dr. Manika Balasegram, Executive Director, GARDP, esteemed delegates, distinguished guests, and colleagues. It is with great privilege and honor that I welcome you all to this significant gathering as we come together to mark World Antimicrobial Resistance Week. This week is dedicated to raising awareness, fostering dialogue, and inspiring action to combat one of the most critical global health challenges of our time, antimicrobial resistance. Before we begin, I would like to take a moment to extend a very special welcome to our Honorable Minister, whose unwavering support, leadership, and encouragement have been instrumental in driving forward our various critical programs, contributing to the national goal of Viksit Bharat 2047. It is under your guidance, sir, that we have seen remarkable progress and achieved several important feats, and we continue to work towards utilizing science and technology to find meaningful solutions for the unmet health needs of our country. As we come together today, we are reminded that India has made significant progress in addressing AMR, but there is still much work to be done. Over the past few years, our efforts have focused on both innovative solutions and practical interventions that aim to reduce the burden of drug-resistant infections and ensure better health outcomes for all. Let me share some key initiatives that are shaping the future of AMR research in our country and actions from the DBT. We have prioritized the development of indigenous and cost-effective diagnostic kits to identify AMR-specific pathogens and developed a repository at the National Center for Microbial Resources, which is housed at one of the DBT Institute's National Center of Cell Sciences in Pune. This repository houses resistant microbes and infective agents for advancing our research and understanding of antimicrobial resistance. Additionally, there are several other key initiatives and national and global collaborations that the department has undertaken, which are aimed at advancing research in this important field. Now, turning to the highlight of today's program, I'm pleased to share some significant advancements in our ongoing efforts to combat antimicrobial resistance. One such milestone is the scientific completion of first indigenously developed drug Nafithromycin. It is a macrolide antibiotic effective for the treatment of community-acquired bacterial pneumonia caused by multi-drug resistant pathogens. Nafithromycin has been developed by Wockhardt, a leading Indian multinational. This promising treatment marks a major breakthrough in our battle against multi-drug resistant pathogens. As we move forward, it is clear that addressing AMR requires a unified multi-stakeholder approach, bringing together government, research institutions, healthcare providers, and industry. As part of the National Action Plan 2 for AMR, being coordinated by Niti Aayog, we are aligning our national strategy with the global best practices. The National Action Plan outlines crucial steps and strategies that will guide our ongoing efforts to reduce AMR in India. Once again, I, on behalf of DBT and BIRAC, extend my heartfelt welcome to each one of you. I also want to express our gratitude to the Honorable Minister for his continued support and his presence here today. Thank you all, and let us continue to work hand in hand in this vital mission. Thank you.
Thank you, ma'am. I now invite Dr. Jitendra Kumar, MD BIRAC, for his remarks, please.
Honorable Dr. Jitendra Singh, Minister of State, IC, Ministry of Science and Technology, Ministry of Earth Sciences, Minister of State, PMO, Ministry of Personnel, Public Grievances and Pensions, Department of Atomic Energy, Department of Space, Government of India, respected Secretaries, DBT, Chairman BIRAC and DG BRIC, Dr. Habil Khorakiwala, Founder, Chairman, Wockhardt Group, Dr. Manika Balasigram, Executive Director, Guard P, Dr. Y.K. Gupta, Principal Advisor, India, Guard P, Dr. Anamika Gambhir, scientists from DBT, my colleagues from BIRAC. I welcome you all to this World AMR Awareness Week. We are going to celebrate this week by launching the soft launch, actually, the scientific completion of the first indigenously developed antibiotic against MDR, isolates Nafithromycin. This antibiotic has been developed by the support of BIRAC through Biotechnology Industry Partnership Program. We are supported with rupees 8 crore for this funding for the clinical trial phase 3. I think the clinical trial phase 1 and phase 2 was conducted in Europe and US, but I think slowly we have to develop those capabilities also. In the morning, we had a wonderful session where a lot of experts in this area had delivered their opinion and thought. And I'm very glad to share that there's a huge potential for fighting against AMR in our country. And I think we need to create this group where all these scientists can come together and, we can have a lot of innovative products. This launch, the soft launch that we are doing, is actually the first antimicrobial resistant drug that is coming out from India, that is after 35 years of any drug that is coming out of the world. So it is a great achievement and a celebration from our side. I think in the first session we had Dr. Lata Kapoor, who gave an overview from NCDC side, and then we had Dr. Anand Kumar, who is also leading AMR innovation program, and he talked on India as a global solution hub for AMR. Then we had Professor Gopal Nath, Department of Microbiology, BHU, who talked on bacteriophage therapy, a magic against AMR. Again, a very good data on how phages can also act as, against microbial and diabetes, this resistance against microorganisms. And he also talked about how it is very important this AMR treatment that we are doing is actually will go a long way to fight against cancer and diabetes also, because most of the diabetic and cancer patients who spend more time in hospitals actually acquire these infections. Then Dr. Bhavatish Das, he spoke about microbial genomics and genomic engineering, key strategies in the battle against antibiotic resistance. Again, a very good work that is being done in our own lab in THSTI. Dr. Aradhana Vipra, head of microbiology, Gangajan Biotechnologies, she talked on bacteriophage product development for non-clinical application opportunities and challenges. Then we had Professor Urmi Bajpayee, Department of Biomedical Sciences, talking on bacteriophage endolysin, promising solution to bacterial AMR with a One Health approach. So with all this, I think there is a great opportunity in India for developing more and more drugs. This drug is very innovative because it's a ketolide macrolide, and I think the keto group which has been added in place of sugar makes it combined to two domains in rRNA, the bacterial 16S RNA, which makes it circumvent against it is circumventing against all the mechanisms of antimicrobial resistance. So with this, I close my talk and I congratulate Dr. Habil Khorakiwala and his team for coming out with this great product, and I wish him best of luck. Thank you.
Thank you, sir. I now humbly request Professor Y.K. Gupta, Principal Advisor India GARDP and President AIMS Jammu, to deliver his address on AMR through innovation and policy.
नमस्कार, आदरणीय डॉक्टर जितेंद्र सिंह जी, मिनिस्टर ऑफ साइंस एंड टेक्नोलॉजी एंड मिनिस्टर ऑफ अर्थ, डॉक्टर राजेश गोखले, सेक्रेटरी, डॉक्टर जितेंद्र कुमार, मैनेजिंग डायरेक्टर बायरेक, डॉक्टर अनामिका, and I see, and of course Khorakiwala Sahab, my very, very old acquaintance, many senior scientists here, and any other media person here. I think this is a historical day, I would consider, when India has developed independently, indigenously, an antibiotic for Indian people as well as globe. This is a matter of great celebration, and I want to congratulate, sir, the BIRAC, who have identified such potential molecule, funded it, and have seen that how this molecule can be useful for fighting what we call today as a possible pandemic, hidden pandemic. Abhi hidden nahi reh gaya. Now this is an apparent pandemic, and it is estimated that 1.2 million population, 6 lakhs population global, 6 lakhs population in India, are dying because of antimicrobial resistance. And बड़ा unfortunate है कि हम अपने ही देश में because of low knowledge of what antimicrobial resistance is, कैसे इसको हम prevent करें, क्या इसके लिए हम treatment करें. 6 lakhs and couple of my friends who are, one of them is dermatologist, one of them was cardiologist, was admitted in ICU. ICU में admit होने के बाद में ventilator पे गए, ventilator से वहाँ पे pneumonia हुआ, and then all antibiotic did not work and they died. When this happens to you, when this happens to your close intimate friends, then you realize where the fault lies. And I would say, in 1970s when I was doing MBBS and sir was also maybe in the same age, antimicrobial resistance used to be a short note question. क्या होता है, कैसे होता है? But today it has become such a magnified thing that right from the school age to geriatric population, people must know it. The important thing is that if we do not know what antimicrobial resistance is, the people prescribing antimicrobial agents at the periphery to secondary to tertiary care hospital, the irrelevant antibiotics, that generates the possibility of antimicrobial resistance. Sir, for the first time in AIMS Jammu, we have started the digitalization of prescription. Right from day one, the first patient entered in OPD, the prescription is being digitalized. First time, and now this is going to be a network of all 22 AIMS. AIMS Jammu will be leader. In this digitalization of prescription, we will come to know that what is the prescription habit also, prescription pattern of different antibiotics, whether rational or irrational, and then the feedback can be given to the prescribing community. That's one. From there, the second thing comes that if you have a quick diagnosis of antimicrobial resistance bugs by molecular mechanism, cheap, affordable and timely, then the life can be saved. Then third thing comes that once you do that what is the policy for bringing out a new drug? The drugs, if you remember, हम लोगों से पहले पूछा जाता था कि Nobel Prize किसको मिला? The first Nobel Prize antibiotics was got by penicillin discoverer Alexander Fleming and Chen. So much so that people thought कि अब हमारी infectious disease खत्म, finished, we've conquered the world. But then soon it was realized that this penicillin, which was recovered from the urine of the patients to be given to others again, it was so precious at that time, it became resistant. It became resistant. And then the discovery of clavulanic acid and all those things, and then support of cephalosporins started working on it. Soon, within 3 years, another Nobel Prize for streptomycin for Voxfen. And that was again considered कि अब हमने tuberculosis खत्म, gram-negative infection finished. That was panacea for all. Now we know that tuberculosis has again a multi-drug resistance problem. Fluoroquinolones, when we considered that when we passed out our MBBS and fluoroquinolones came and we thought that now this is the end of the infection. Now fluoroquinolones, 90% of the fluoroquinolones are resistant. Race between the bacteria and race between the microbiologists and the pharmacologists, or you can say chemists, continues with these bacteria. And who is winning the race? Clearly, the ahead of line is bacteria. We are falling short because the pipeline of antibiotics is drying. The receptors of the antibiotics where it can be attached are almost exhausted. Industry is not interested to put money because they find that the return of investment is uncertain, because unlike anti-cancer drug, anti-diabetic drug, anti-obesity drug, कि वो एक बार लो, then this will continue to be given for lifelong. The return of investment is unlimited. If you give antibiotics, then the return of investment is limited 7 to 10 days therapy. and that too, if it doesn't work, then you have to shift to other. And then the most important thing is, if this precious antibiotic which today we are celebrating, or tomorrow we will develop, if this antibiotic falls into the hand of a monkey, if you give a very sophisticated digital camera or digital instrument in the hands of monkey, what it will do? It will break it. And that's why we say, when these specific, so life-saving, precious antibiotic is used by irresponsible, I would say, not incompetent, not illiterate, irresponsible physicians, irresponsibly to where it is not required, 75% of the antibiotics globally, not in India, globally are used where it is not required. Only 25% at the places where it is required to use. Indian figure is unfortunately not available, but it cannot be different than what is global. So now if this goes in the hand of the monkeys, means the irresponsible physicians, then again this tragedy will happen, that this antibiotic will again go into the resistance pattern. And therefore, important thing is, policy has to be that how we can reduce this irresponsible use, whether through education, stewardship, or regulation. We have a regulation, as I always say. Making a regulation, sir, is easy. We have Schedule H, we have Schedule H1. Now we cannot make Schedule H2 and H3 or H1 A, B, C. We have to have a policy of training, policy of enforcement, and policy of continuing education to the public, to the doctors, and to the chemist who gives the antibiotics as an OTC medicine, free of no need of any prescription, that contributes to antimicrobial resistance. Lastly, I would say, I co-chair the this DBT's One Health project. It's not the only problem which is limited to human. It is problem which is from human to animals to aquatic to environment. Now, if the animal use of veterinary, which is not so much controlled in India, though there are several regulations have been prescribed, it goes into the wastewater. The number of only few studies where the wastewater experiments or wastewater sewage estimation of antibiotic has been done. It comes back to the human and is a possible cause of resistance. In environment, in earth, the traces of antibiotics can be found. The irresponsible discharge of sewage from pharmaceutical industry, the irresponsible waste of unused medicine from hospitals, incineration is a guideline, but many times incinerators are not used, particularly in secondary and primary care hospitals. So this creates an ecosystem which is conducive of antimicrobial resistance development. And therefore, sir, since you are having a couple of ministries together and you have a lot of influence of other ministries, why not we create a comprehensive policy under One Health program, as well as antimicrobial resistance development? So we have to have 2-3 policies here. One is sufficient funding, sufficient cooperation with the industry and academic institution. As I said, no industry is interested to put money. I congratulate Khurakiwala ji that they desired and they worked it and they took risk on that. Return of investment and the high risk, de-risk the investment on that. Give liberal funding and make a strict policy through Ministry of Health or through Ministry of Environment that those who prescribe antibiotics, those who dispose antibiotics irresponsibly, are equally punishable as committing any serious offense about the drug regulations. So thank you very much, and again I congratulate BIRAC, I congratulate Department of Biotechnology, and I congratulate the WOCAD, and of course I compliment Dr. Manika and his team for GARDP, who is working with the objective to bring an antibiotic or antibiotics not only in low and middle income countries, but primarily at an affordable cost, because the drugs which are very effective, few drugs which are coming, they are so expectedly, so exorbitantly high that it will be very difficult for any government to support it or the patient to support it. So unless they become at affordable cost, I think it will be, this will be an important issue which we have to be addressed. So thank you very much, sir. Thank you.
Thank you, sir. We truly hope that these small efforts have a big impact in creating a more antibiotic responsible and AMR conscious world. Next, we have the privilege to have amongst us Dr. Manika Balasegaram, Executive Director, Global Antibiotic Research and Development Partnerships, who will be discussing the role of global perspectives and unified talks in addressing antimicrobial resistance. Dr. Manika, please.
So first of all, thank you very much for inviting me here. I would like to give my thanks to the Honourable Minister, to Dr. Rajesh Gokhale, the Secretary of DBT, to my dear esteemed colleague, Professor Gupta, as well as Dr. Jitendra Kumar from BIRAC, and my heartfelt congratulations to Dr. Korikawala and the WOCHAD team. This is a remarkable achievement, and I think will probably be the first of some more achievements to come in the future. So I'm here to represent GARDP. We are the Global Antibiotic R&D Partnership. We are a non-profit organization and foundation. We are based in Geneva, but with team members across the world, including a team here in India. We partner with both the public and private sector. We're largely public funded. And our objective is to develop and ensure the early access and affordable access of new and improved treatments to address this growing problem of antimicrobial resistance. And we have several projects in our portfolio, and I would like to point out that we work with several partners here in India and work across, I would say, the life cycle of research and development with our partners. So, for instance, we are working with a small biotech based in Bangalore in C-Camp called Bugworks. Very, very exciting project, very early stage in early clinical development, and promising to be a treatment that can really address multi-drug resistant gram-negative infections and gram-positive infections. We're also partnering with Origin, which is part of the Dr. Reddy's group, and Origin has been an extremely important partner in helping us to develop a new chemical entity to address multi-drug resistant gonorrhea, and this treatment has actually finished phase 3 trials and is now proceeding for filing and approval. And we also have a third project with another company called Orchid Pharmaceuticals that is really actually helping us to develop a lower cost, quality assured formulation of a recently approved drug called Cefiderocol. And we're intending to bring this treatment to India and to patients across the world. So I would probably make one key point, which is to say that I think we're living through a very exciting moment in some respects. We have a great problem ahead of us with antimicrobial resistance, but a huge opportunity in the future, particularly here in India. And I think there's an opportunity to really turn the narrative on its head, to look at really developing treatments and developing new treatments and doing clinical trials here in India. Also to look at manufacturing, quality manufacturing and supply, not just for India, but for the world. And also to look at early introduction and access in India as a pathfinder country for access, but India also representing a considerable part of the world's population and therefore actually a market for antibiotics in the future, albeit with, as Dr. Gupta, Professor Gupta mentioned, close stewardship and monitoring. And I think these three important components, the ability to do world-class clinical trials, the ability to do world-class manufacturing, and the ability to introduce these new antibiotics first and foremost where they're needed the most is, I think, key on how we're going to address this problem of antimicrobial resistance. And this is something that I believe India will be a leader in the future, is already a leader in today. So I will close my remarks by giving my thanks again to all of you and to the Honourable Minister And again, a big congratulations to the Wokhard Group. Remarkable achievement. Thank you.
Thank you, sir, for sharing a glimpse of the efforts of GARDP in combating AMR. I now invite Dr. Habil Khurakiwala, the founding chairman, Wokhard Group, to share their journey for development of the indigenous AMR drug, Nafithromycin. So Wokhard has a video to play, which will be followed by his presentation.
Curiosity, humankind's greatest attribute and its greatest superpower. It helps people discover things no one else knows about. Takes them to places never thought possible. Teaches them about the world they live in. Curiosity makes people human. Knows no age. And propels mankind forward. One person's curiosity helped us discover gravity. Another person's curiosity led to the invention of the telephone. A third person's curiosity helped invent the computer and the internet. But what if curiosity had a higher purpose? What if it could save millions of lives? 25 years back, a team of scientists led by Chairman Dr. Habil Khurakiwala got together and put all their passion, focus and commitment on a global health challenge: antimicrobial resistance. Antimicrobial resistance costs 5 million lives annually. Jim O'Neill, the famous British economist, has theorized that this number could increase to 10 million a year by 2050. And multiple times that, the number of hospitalizations. Team Wokhard filled the breach left by the world's top pharma companies. They scaled up their internal competencies and invested $500 million over 25 years. Created a culture of research and innovation, and made it India's first and the world's most innovative company in the antibiotic space. Research is just formalized curiosity. So, Team Wokhard joined the dots and discovered gaps in the global pipeline of drugs. They conducted research programs tailored towards various community needs. The spirit of curiosity sparked a huge interest amongst the infectious diseases scientific community. Eminent experts, universities, think tanks and global opinion leaders went out of their way to partner Wokhard. And because the drug development was de-risked from a very early stage, they didn't face a single failure at the clinical trials. The work was featured in more than 200 top-rated international publications and conferences globally. Curiosity and research leads to new knowledge. So Team Wokhard developed a burst of 6 new drugs, 6 new NCEs meeting the US FDA and WHO unmet needs criteria. The leading one is Zenic, the others being Miqnaf, Enroc, Enroc-O, Forisco, and Odrate. Zenic is a one-of-its-kind antibiotic discovered in the last 100 years. It's been designed to fight the major superbugs in the world and prevent as many deaths as possible happening due to antimicrobial resistance. Zenic has already saved more than 35 critically ill patients in India and the US under compassionate grounds when all other treatment options have failed. Curiosity is a never-ending journey, and Wokhard is here to enable it. For you, for life. Wokhard. Life wins.
Very good afternoon to all of you. Honorable Minister Dr. Jitendra Singh, and in the fact that I just was talking to the secretary and he mentioned that 9 secretaries report to you, and I have been involved with India and the government for many, many years, and I have rarely come across such a wide range and array of portfolio you are handling today, sir. So it was indeed very happy to meet with you personally today. Secretary Rajesh Gokhaleji. And then we have Dr. Y.K. Gupta, who has been involved with us, and Dr. Jitendra Kumar, the MD of BIREC, and the scientists and distinguished members who are present today, and friends. What I would like to share with you is the story of WOCAT. Over last 25, 27 years, and the journey which we undertook to create an organization for a drug discovery in a competitive world. Because when we talk of science and the new drugs, we look purely on that basis, but we took a view that you can only have a new drug worthwhile provided you understand global competitive scenario of what is happening. And we found that when billions of dollars are being spent by a large pharmaceutical company, a small company like ours in 90s have no resources, both financial nor scientific resources to compete. So we had to find an area where big pharma is vacating because of reasons already mentioned earlier. And we saw a 20-year trend that antibiotic research by big pharma are declining, and approval by US FDA was year to year declining for 20 years. And we said the organism requires that they develop a resistance and you require new antibiotic. And India and emerging market had about 10, 12, 15% of total value business coming from antibiotic. So we thought that there is a chance that if we focus on antibiotic, the chances of success are high because we don't have a competition, and therefore we took this journey forward. Hello. Yeah. So that is how we really focused on antibiotic research. And over the years, the burden of anti-infective and resistance are increasing. And that is the focus of our antibiotic research. Just to get you a problem, which we just saw, there are 5 million people who are dying today. And if the prediction are there, it will be 8 to 10 million by end of 2050. But we believe these are the prediction without WOCAT presence and WOCAT work in antimicrobial research. It's only one drug, but we have a series of drugs coming in. And we believe that 5 million should be less by 2050 rather than the more one. The next, I think this we already saw in the slide that we have about 6 of our molecules. It is a 25-year work basically. And we have a team which is led by Dr. Mahesh Patel 25, 27 years back. He is the chief inventor of all the new molecule we have. Today he is a chief scientific mentor in the organization and very ably led today by co-inventor Dr. Sachin Bhagwat, who is a chief scientific officer. And I must like you to recognize both these people and the entire team for a tremendous work done. There is one aspect which I must like to emphasize here, since our minister is present also, is US and many countries wherever there is a major unmet need, they have developed a fast track method of approving drugs. Like all of our 6 drugs which are in the various stages of development are given that fast track. They call it QIDP, Qualified Infectious Disease. So I believe that in India, where our problems are there, from a regulator point of view, we must focus on some of the India's problem and develop this kind of a category so various regulatory bodies respond very quickly to get the drugs approved, basically. Now, the other thing which I would like to emphasize here, sorry, I'm not very good with this stuff, that, yeah, the important thing is that to find a drug discovery, there are 15 different competency we have to build up, which is mentioned here. Now, to build up this different competency, this is a creation of a new organization and building up various competency like microbiology, toxicology, safety, pharmacology, and so on and so forth. So, this is something which takes time and it's not easy to develop for anybody. And actually, we learned over hard way and took us about 8-10 years to build up these competencies within the organization. And then we got into a space where we found a lot of new drugs. So over the years, we have screened 8,000 molecules and we have filed a global more than 400 patents. We have conducted 25 phase 1 studies, then phase 2, and 3 of the phase 3 studies are completed and some are undergoing. See, in order to create an antibiotic research organization, one of the many years back, what our leadership, Mahesh and the team did is instead of getting experienced people with 5 years, 10 years, 20 years experience, the entire team was created by directly from graduates, postgraduate and doctors from the universities. So what they focused really on the technical competence of the people required. And they had a fresh mind, they had an open mind. You can mold them the way you can mold them. And I must tell you, over this 25, 27 years, every organization has an attrition. We hardly have any attrition in my research program over 25 years. And I know many scientists work their whole lives and they don't find a drug. So our team is highly motivated because it's not one drug, but there are so many drugs they have discovered and one after another they are getting launched. The other important aspect which I want to highlight here, no single organization it is possible from an Indian perspective to do everything. So what we have done over the years, we have about 30 global institutes where we have interacted from time to time. And the competency gap which we don't have and they had, that is getting filled. Like for example, for a drug for approval in the Western market and everywhere else, how do you say an antibiotic is good and it is multi-drug resistance, is you have to regularly screen all the strains of organism, and these are there are organizations who do that in US. So, and FDA or regulator in Western countries do not accept data over 3 years. So, whatever you want to prove, you prove that your drug is effective of currently available strains of the organization, and then the drug gets approved. So, this kind of competency, I just give one example. There are so many other areas we require, and therefore it is a network of these institutes which are universities and research labs worldwide which we have used. So what is the result today? Today we have 6 drugs, antibiotics, which are already discovered. Out of that, 4 of them have completed phase 3 clinical trial. First 2 are introduced already. Miqnaf is the one we are discussing today, Nafithromycin. Zenic is another important drug, gram negative, which I'll talk to you a little later more about it. And the other 2 are in the pipeline of various stages of development. One is in phase 2. The other one, now I must tell you, in this WCK 6777, actually NIH, US, National Institute of Health, funded our phase 1 clinical trial, and they did it. And they spent $9 to $10 million because they felt that in US you need this kind of a drug urgently. So that is the kind of work we are doing. Now let me just share with the Miqnaf, which we are talking about today specifically. It is basically, as we all know, is a community respiratory infection. It is an oral drug. They say there are about more than 2 million deaths because of pneumonia. And in India, the 23% of global community pneumonia burden is here in India. And these are the pathogens, typical, it works both on typical and atypical organism. And no new antibiotic has been developed in this space anywhere in the world for last 30 years. And the one which are currently used extensively, like azithromycin, have 60% resistance in India. Then amoxiclav combination doesn't work on atypical organism. So for practical purpose, the doctor somehow manages to treat a patient, and if they can't be treated orally, they go to the hospital. So about 1/3 of the pneumonia patients are hospitalized today. And therefore, what is the uniqueness of the molecule? Very, very briefly, it is 10 times more potent than azithromycin, which is the last drug discovered. The lung penetration of nafithromycin is 8 times more than azithromycin. So if you multiply these two, that is a delta difference of power of nafithromycin, and therefore it is only used for only 3-day treatment, and it works. Because we did a phase 2 clinical trial with 5-day and a 3-day treatment, and at the end of the clinical trial we found that both gave equal results. So obviously you choose lower doses rather than higher doses of antibiotic. And it has hardly any side effects, high tolerability to gastrointestinal, and it's not affected by food. There are no drug-drug interaction. So from that point of view, it is a very safe drug. It can be given, and that is the drug we are talking about. Now, phase 1 and 2, which we did study in the US and Europe, basically, and I was discussing with our secretary earlier that why do we do phase 1 outside India and not in India? Main reason is time. Because of our process, the time it takes to do a phase 1 clinic, even though it costs us a fraction of what we spend outside, is killing actually. It takes more than double the time. Outside you do in 8-9 months, it takes 1.5 years to 2 years to do it in India. And for a new drug discovery, unless you do phase 1, you can't move further also. So from that point of view, and that is where I think we should look into it to simplify and bring the global standard of doing various stages of clinical trial here. I would like to thank BioRec. They participated in this program and give us some funding in phase 3, not fully what we needed, but a part of it. But whatever was there is welcome. But just to get you a sense, we have spent collectively 500 crores to get this drug out right from the beginning in research. Now, similarly, we are having another molecule 4282, where BioRec is looking at funding. Let me talk to you on another important drug which would be introduced in India, and probably it will save millions of lives. And that is our WCK5222 Sidabactam Cefepime combination. The brand name we have given is Zenic. The carbapenem resistance associated the mortality, what you see is a tip of iceberg today. And nearly 9 lakh mortalities with gram negative happens every year in India. And 56% of mortality during COVID-19 was there, and which resulted in about 10% the patient who passed away. And there is a 30% resistance to the main drug which is available today, meropenem and others. The important aspect is of the Zenic or 5222 is it is a new class of antibiotic. Like you talked of cephalosporin, the quinolones. So it is a beta-lactam enhancer. And therefore you see a very unique effectiveness of this drug. And bacteria is not used to look at the action what Zenic provides or 5222 provides. Second, another important aspect I must mention, CLSI assign a susceptibility to an antibiotic, and it is a global body, and when you do a diagnostic test, normally the higher the susceptibility, the more effective is a drug. So in the last 15 years, many drugs have received 4 to 8 breakpoint. Our drug, they have assigned 64 breakpoint for all the 10 in organism where it is effective. This is such an important milestone in antibiotic research that this is, we call it a penicillin moment. 100 years back when penicillin was discovered, it opened up a new vista. So a drug which works, and no antibiotic in 100 years have been given 64 breakpoint for 10 of these infection. So from that point of view, and that is why this drug is working so well. We have more than 100 international publications for these drugs. We participated in conferences, there are publications in journals, and there are poster presentation also. A very unique thing is that when a drug is not approved, which is not, this is not an approved drug today. So what happens, if there is an idea in India or worldwide, a compassionate use. So and when everything do not work, you go to the drug controller and give the complete evidence of the patient and say nothing is working, we want to try this drug. So I must compliment the Dr. Dubey from Medanta, who was the first, based on literature, went to drug controller and got the permission, and since then we have saved 39 lives. And these are in bacteremia, HAP and VAP pneumonia, meningitis, complicated intra-abdominal, and many others. And in some of the bone infection, where it is, or meningitis, where it is difficult to treat, a longer treatment, normally it is recommended 7 to 10 days, 15 days. But their doctors have given up to 70 days. And the patients have been cured and no side effect. And we literally have 100% outcome on this compassionate use of 39 patients. Out of that, there is a US patient where US FDA has also cleared a drug for compassionate use. This is the University of California, Irvine Medical College. And that patient also was given 30-day treatment. There is a one going on again in another US patient, similar kind of thing. And all these bodies worldwide, FDA, AMR, Cisco, have given a fast track like US FDA QIDP status to the drug. This is just to get a competitive scenario which I mentioned at the beginning, how you become a competitive in research. And this demonstrates that we have 6 drugs at various stages of development or approval of phase 3 completed trial, whereas all the other companies, either they have 1 or 2 drugs. Some of them are not their own research, they have bought it from somebody and they have introduced the product from here. So that is where we are the most successful antibiotic drug discovery organization in the world. And I think not only we should be proud, India should be proud. And we have an ongoing program over the years to maintain this leadership in years to come. I have a small request, since the minister is here and the secretary is also here. So what our suggestion is, that there are a number of drugs where a GST is exempted, like human blood, component type of contraceptive, or a 5% GST is there for insulin, cancer drug, cyclosporin, ORS, hepatitis vaccine. My request is these antibiotics which are discovered in India must receive the same kind of a status, because ultimately it will become more affordable to the patient. And this is an urgent need. This is a need which saves lives. So from that context, this is an appeal. And I just want to close by Nobel laureate Govind Khurana, what he said, the pursuit of science is not to be just pursuit of knowledge, but also pursuit of better and more humane world. Thank you very much.
Thank you, sir, for sharing with us WOCAD's journey to where it stands today. I now invite Secretary DBT, DG BRIC and Chairman BIRAC, Dr. Rajesh S. Gokhale for his address, please.
Namaskar and a very good afternoon to all of you. Honorable Minister of State, Independent Charge, Minister of Science and Technology, Dr. Jitendra Singh Ji, Dr. Habil Khorakiwala, who so passionately talked about his work now on founder chairman of WOCAD Group, Professor Y.K. Gupta, who is also the chairman of RCGM, which runs through the DBT actually, I think, which is one of the regulatory aspect of looks at this part of this. He's been a formal professor at AIMS. Dr. Jitendra Kumar, MD BIRAC, Dr. Anamika Gambhir, advisor DBT, speakers of the morning session who so eloquently talked about their innovative ideas and R&D work that is going on in different places. My colleagues from DBT, BRIC directors from BIRAC, honorable guest, including Dr. Manika, who is here. Thank you very much for being here today, actually, and his team. And I think it's a wonderful day to celebrate success on a week which is actually highlighting the MDR all across the world. I think we should congratulate Dr. Khorakiwala and his team for fantastic work they have done over a period of time. I think it's been fantastic for BIRAC to partner with such wonderful endeavors over the years, and we've been doing supporting various innovations in the country. And Department of Biotechnology, after it set up BIRAC, has done this fantastic achievement role of not only supporting startups, but through various missions such as National Biopharma Mission or various other programs, have provided various aspects of biopharma missions that have taken India into the next forefront of frontiers of how innovation can be deployed. I think I always believe that innovation ecosystems are of 3 kinds, and one, as I always like to talk about the disruptive innovation, and this is what Dr. Khorakiwala talked about today, is the disruptive innovation. It really changes the course of the time of how these will actually work together. Often we do a lot of sustainable innovation in the country, and even more what we do is efficiency innovation, which actually is a little bit of a
improving our own activities. And I believe Indian companies have to grow up to the levels where we actually start to do disruptive innovations even at a pace that is unprecedented, primarily because we have enormous amount of talent in the country. We now have a supportive environment with the government. Actually, an honorable minister who is here today is a wonderful predicament for his commitment to take forward all these wonderful activities in the country. And also, there is will, even in the regulatory perspective, to improve the quality of regulatory work that is going on in the country, which is a fantastic sign, I believe. And I think that's where we should credit our honorable minister for taking up all the interest in these activities, because he not only is a very passionate guy, he's being a clinician himself, he loves this areas of innovation, and having done several activities, or including, had trained PhD, MD students while he was as an academician and a diabetologist. I think his guidance is very, very helpful to us. Thank you very much, sir, for giving time. And your support has been absolutely fantastic to Department of Biotechnology. I think as you would really understand the problem of various people have talked about, the AMR transcends all the borders, discipline and sectors actually. It doesn't matter what disease you are suffering from, any chronic disease, eventually weakening of your immune system causes to have an antimicrobial problem actually appearance to do. So it's kind of not only specific to this one specific sector, it's really thematically, as Dr. Y.K. Gupta very nicely said, is drug resistance is not a destination, it's a journey actually. You will have to continuously come up with new molecules, new ways of finding mechanisms, because any new drug you put out in the market, you are likely to develop resistance to it actually. And of course, the indiscriminate use will create craziness. I wouldn't even like to call drug resistant. It will cause craziness in the world. And we will have to keep coming up with innovations that are at a very, very rapid pace. I think the completion of phase 3 in India is a spectacular success of Nafithromycin. And as Dr. Korakiwala actually very nicely talked about, that why it takes time to do all these activities. But as he's shown the portfolio, so we are very happy to see that you have 6 molecules coming up actually, and we look forward to its very, very rapid deployment in the country. And DBT BIRAC will be more than happy to help you out in any endeavors of putting together these activities as much as forward as you think the Western world is doing as well. Believe me, the immense support the government is right now providing to SNT institutions and innovation has been unprecedented actually. I think that's why you can see the startup ecosystem that is growing up like amazing, like anything. I mean, there are more than 1 lakh of startups now. And in fact, in BIRAC, since the BIRAC formation, there were about 85 startups before this, and now about 8,000 startups in the country. So it's amazing what we're able to do in this country right now. And it'll be amazing what we will be able to do it in the future. I think the other aspect that India should always be proud about is the cost of the treatment that we provide to a patient in this country. Actually, I think that's something that we never celebrate in this country, which I think we should be really, really very, very thankful to the government policies and mechanisms to look at. I mean, if you really look at the way, we're proud of how US does innovation actually. But if you really look at what you do, US innovation model is, what ideally you want innovation to do is to bring more effective, more efficacious, and more affordable drugs. Because the affordability is the key for the populations to actually be able to accept these aspects of it. And I think that's not what you observe nowadays actually. Every single drug that is coming into the market is expensive than the previous one. And that tells you that the models probably need to be reworked with. And believe me, India is a destination if you want to change this perspective of the world. Every country today is looking forward to India, how India will innovate, how India will take forward any of these activities. I mean, the examples of very many, for example, forget the drug, I mean, the more recent success of financial management ecosystem of our country, of how UPI has shown the way. If you would have followed the international model of using credit cards for every transaction that you follow, what government did was set up the DPI ecosystem in the country, digital platforms, which is actually free of cost to everybody. So you can see the financial transaction model in the country is quite absolutely stunning which has actually stunned the entire world of how transactions can happen without somebody making money in the business. And that's what the power of this country is, believe me, is to bring the best possible technologies, best possible innovations, and make sure it happens for the people and not just for the, for some companies to make money out of it. I think that's why Nafithromycin, again, is a classical example. It's an ultra short course therapy. Again, outstanding in its own ways, 3-day course therapy actually. And obviously, as you mentioned, the concentration of lung concentration is very high actually, despite its very short therapy, course therapy. And I think that's what makes very, very important monotherapy for community-acquired bacterial pneumonia. So it's a giant stride that we are taking today. And I think this is the combination of, I like to call, ladies and gentlemen, of vision, innovation, and determination. Of course, you realize it takes years to build all these capabilities, and also as he mentioned about the team members, about different aspects of 15 different departments that he talked about actually. These are all very, very important, and I think the ecosystem will just likely to grow, and in fact is going to be taking a completely different trajectories in future to look at. I think I will stop here, but again, congratulate each one of you for your endeavors. I think Department of Biotechnology has been driving innovation, capacity building, and entrepreneurship, and now talking about bio manufacturing as a way to create value in the systems. Of course, before ending, I will also talk about a couple of interesting programs. I think you mentioned about the biorepository that has been set up for microbial resource. Actually, there's a very important program going on neonatal sepsis actually as well. And I think it's again a fantastic program that is looking at how neonatal sepsis is caused in young children and how this can be really managed. A really fantastic comprehensive etiology, transmission, and severity of neonatal sepsis is being worked out in an Indian context. Similarly, there's a program on genome sequencing of Mycobacterium tuberculosis genomes. Actually, if you really look at presently the drug resistance, actually to identify drug resistance, it takes about microbiologically, it takes about 3 weeks to come. If you want to identify which drugs are resistant, and obviously by that time, you cannot go into the drug, the therapy that is the second line treatment. Now, can you use genome sequencing as a way to be able to analyze the data very, very quickly within 5 to 7 days and be able to put people on the right kind of medicine that they are meant for? And I think these are the kind of important R&D activities that are going together, which will again transform the ecosystem and will create momentum to what I call ideate, innovate, and implement solutions for the entire country. I congratulate BIRAC and Wokhart team for this important milestone, and I eagerly anticipate future successes to improve lives and shape the better tomorrow. Thank you very much.
Thank you, sir, for those enlightening words, and we are confident that the department will continue to thrive under your able leadership. I now humbly solicit our chief guest for today, Honorable Minister, Dr. Jitendra Singh Ji, to formally announce the scientific completion of this indigenously developed antibiotic Nafithromycin. May I also request Dr. Sanjay Saxena, Dr. Sonali Tandon, and Dr. Prachi Kaushik to join them on the dais for a group. Thank you, sir. May I now request our Honorable Minister, Dr. Jitendra Singh Ji, to please address the gathering.
Secretary, Department of Biotechnology, Government of India, Dr. Rajesh Gokhale, who besides being the secretary also happens to be a noted researcher, scientist, and one of the leading biotechnologists of the country. With a lot of new ideas, innovative ideas and passion, as you would have realized in the brief discourse that he made. We have besides us Mr. Jitendra Kumar, the head of BIRAC, and I must compliment him that with his perseverance, BIRAC has emerged as a kind of a role model and example of the successful public-private participation initiatives. I think the only other such instance I can think of is in space, that we have also established to promote PPP models in the space sector. And I'm glad that I happen to be associated with both the departments. Dr. Y.K. Gupta is a veteran, in fact our senior in the medical faculty. He retired as the head of Department of Pharmacology, KG Medical College, Lucknow, so very familiar in our circles. And after superannuation, he's also been active still. And I have to share with the audience in this room that under his leadership, the AIMS at Jammu has emerged as one of the most state-of-the-art, latestly equipped AIMS among the country. So the other AIMS are now following the manner in which the technology has been appropriately used. It is to be seen, and I in fact have suggested to some others also in the hospital administration to go and see the model, because once the patient enters, he gets a print on his mobile, so he doesn't have to ask which way to go. So he is guided by his mobile to whatever OPD or department he has to go. And beginning from there is just one entry pass. And of course, Dr. Khorakiwala, he'll be amused to know that though I have met him today, but WOCAD has known me for several decades. The problem of, or maybe the peculiarity of being a successful practitioner is that you don't have to deal with the proprietors. The managers take very good care of you. So for many years, I was a darling of their HRD division. And I've also, it'll be a revelation to you, and of course, may not be even to you. Notice that I have availed of the services of your guest house in Mumbai. I think that is Juhu Manzil. Yes. See, I still remember. So in my younger years, I have stayed there for long spans of time. It's just opposite Amitabh Bachchan's house. That is Pratiksha or whatever. Jalsa, I think. So, and in fact, when the Leelavati hospital newly opened, Mr. Mehta opened it in the name of his mother, Leelavati. It's possibly one of the first reclamation constructions on the seaside. We were young then. So in its earlier weeks, I had to spend some time there. We were conducting a workshop with Novo Nordisk, of course, in my own stream of diabetologies. And again, I was your guest. And this was located in Bandra and this in Juhu Manzil. So we used to travel in the local train. And the night train we took, we had all the extras from the film industry singing all the choicest songs to keep us company. But then later on what happened that my practice increasingly got concentrated on diabetes. And almost 99% of my patients were diabetics, and even when I came to the government, I had a waiting consultation of more than 3 weeks. And WOCAD, incidentally, was not very much deep into anti-diabetics. So gradually they started losing stake in me and I lost stake in them. But I am glad again we have found a meeting ground and we have found that there is a reason for each one of us to relate. But I have to compliment you with the sustenance with which you have maintained the house for over half a century. Now, as far as the Department of Biotechnology is concerned, while of course complimenting you for this formal launch, I must say that this is one of the better times happening for the Department of Biotechnology in India. Also because India has a department which is among the better ones in this part of the world. We have conducted trials which have been first of its kind, whether it's the HPV virus, or the DNA vaccine. And also because we have the secretary, as I said, who before being a secretary happens to be a passionate biotechnologist himself. And the remarkable achievements that have been happening in this department over the last few years are also because of the fact that the department did exist, but what was happening here was either went unnoticed or even underrated. And primarily there were three reasons for that. A, that there was very little awareness about biotechnology till a few years ago, and if you asked a common man walking in the street, he would not be able to figure out whether it was biology or technology, engineering or medicine. Second, there was not much of prioritization from the political dispensation, which has happened after Prime Minister Modi came in. He is himself taking presentations, as Rajesh would bear me out, even before the Bio E3 policy came, a series of presentations taken by PM himself, which I don't think happens anywhere else in the world because the head of the state doesn't have time enough, and trying to improve upon the presentations each time. And third, of course, because the scientific experts working here were always shy of publicity and shy of media, which I have succeeded in pushing them in the last 2 or 3 years. But actually, I would say that things began to change after 2014, and that's because, as I said, Prime Minister Modi gave high prioritization to all the scientific ventures, and therefore biotechnology department also had the opportunity to explore its potential. And as I said, the presentations preceding Bio E3, similarly you had during the COVID times, I think virtually every day there was a group which was making a presentation, which also included Secretary Biotechnology Rajesh, who would be directly face-to-face updating the PM and taking his pulse and also discussing things. So that's made a lot of difference. The result today is that, as he was mentioning, we have more than 8,000 biotech startups, but going by the theory of relativity, because that's what makes the difference, we just had 50 before 2014. So the point important to be noted is that we have from 50 gone up to more than 8,000, nearly 9,000 in the last few years. And our bioeconomy was just about 10 billion US dollars, which is now about more than 130 or nearly 150 billion dollars. They're looking forward to the projection of 300 billion dollars. And that's also important because we have to go around and tell people that the next industrial revolution is going to be essentially around bioeconomy, like the one which happened in 1990s, which was mostly IT related. The next one is going to be mostly biotechnology related. And the work on that has already started on a big scale. And I think the golden moment for the Department of Biotechnology and all the biotechnologists of India, may I say with tongue in cheek, was COVID. Now, what came as a curse for others came as a virtue for the biotechnology department. So every adversity is accompanied with. It was during COVID actually that the public awareness about biotechnology awakened. As I said, till a few years ago, people wouldn't figure out. But then you saw every anchor in television turned into a biotechnologist. So they were discussing vaccines, they were discussing these minute nuances of biotechnology to whatever extent they understood. But at least they pretended that they understood. So to pretend that you are aware is also an indication of your passion to seek awareness. So there was also an urge to learn about what it was about, because when you are faced with the threat of death, you are ready to do anything. So COVID helped us to make a quantum leap, and I am glad that this department lived up to its responsibilities. We were the first to come out with the DNA vaccine, which was also provided to almost 30 countries, or more than that, across the world. The HPV virus vaccine, which is popularly got to be known as the vaccine for cervical cancers, but of course Dr. Y.K. Gupta would bear me out. It's not per se anti-cancer, it's just antiviral. A virus which happens to be one of the factors in causing cervical cancer. And since cervical cancer is very rampant in India, so it's got to be synonymous with that. And that's also the secret behind is becoming more and more talked about. And so what was happening earlier was that whenever something came out from the Department of Biotechnology, all the industrial house manufacturing it or marketing it would get away with all the accolades, profits, and even awards. Whereas the scientists working behind would get unnoticed. And therefore, I think it was a year and a half back, I suggested to Rajesh, now it's time you should start blowing your trumpet. Because at least in India, nobody blows others' trumpet. तो आपको अपनी पीठ खुद ही थपथपानी पड़ती है. दूसरे तो समझते हैं कि आपकी पीठ बैठ जाए या झुक जाए. And I told him, see, the DNA vaccine, all the credit has gone to the house that has come out. So also the HPV. And the common man makes out that it is entire their baby. It's just like you watch a good hero and a heroine on the movie and you forget who's the script writer. But you are the script writer, so you just try making noise after every successful trial. And I'm glad today that he is sharing the platform with Dr. Khurakiwala before Dr. Khurakiwala could have gone it solo. Which of course I don't grudge him, because that's part of the professional requirement. I mean, a good industrial house had to publicize itself, otherwise how is we going to sustain such a huge industrial empire for over half a century? So he would do it, he has to do it, he's expected to do it. If he doesn't do it, it would be otherwise. But for the kind of recognition that our scientists deserve, you have to do it yourself. And as he was mentioning, I'm glad that besides the vaccine, they have also been able to identify some of the antimicrobial resistant pathogens in the recent years. A very remarkable exercise that he mentioned about the Genome Surveillance Consortium for Tuberculosis, having identified more than 30,000 mycobacterium strains. I think from the India's point of view and also from the flagship programs that we are being pursued, including TB Mukt Bharat, that's a huge contribution, and also very much in keeping with what the PM often keeps retreating, whole of government, one government. So this is one example. He complementing the efforts of the Health Ministry, the Environment Ministry, and the government as a whole. And they have also prepared a priority list of the Indian pathogens. And as he mentioned, partnering with the global R&D hubs. So now, of course, today is another breakthrough of this Nafithromycin, for which the discovery has spanned over 10-15 years. And as he rightly mentioned, Rajesh's ultra short 3-day regime, which will be much more compatible and amenable to a patient's follow-up compliance treatment. And for that, again, I have to compliment BioRac and also WOCAD for having partnered that. Some of the issues have been raised by Dr. Khurakiwala, very rightly so, about the competencies. I think we are moving in that direction, and I agree some of our trials have to be conducted out that because we don't have the facility enough or the milieu or the ecosystem for that. But gradually now we are trying to live up to the global strategies, global standards, and that's how we can achieve also the goal of Viksit Bharat. So we are very consciously, very seriously seized of that. About the regulatories, and as Rajesh rightly responded, there has been a tremendous improvement in the last few years. In fact, because I'm also dealing with the Department of Personnel and Training, gives me an opportunity to share with you that at least not less than 2,000 or more regulatories have been done away with. So we are trying to shorten the procedures, also try to seek single window culmination of the processes, and thus the gestational period is on the decline. And I'm sure if we have to compete globally, we'll be at it, and we'll be able to achieve the global standards very soon, sooner than later rather. And about GST, the lesser said than done. Because if I was in his place, I would have said that. But if he was in my place, he would have not said that. So you can understand that. And not fully to his satisfaction, there has been a remedy dug out by the government, by Prime Minister Modi's idea itself. We have now started generic kendras, Ashudhi kendras, Jan Aushadhi Kendra, which will be setting Nafithromycin, not Miqnaf, which will be selling Nafithromycin at much lesser the cost, but it might not be Miqnaf or Sivokar. But still, the market will be, the cake is too big. So I'm not sure whether the GST can be that issue can be taken up that easily by the government. But nevertheless, I believe as we move on, as the market dynamics I have experienced over so many decades, the cost comes down. Like it has happened in case of every other antibiotic, whether it was ampicillin. There were times when I was, I think, an undergraduate, ampicillin was one of the latest, and it was available only in Mumbai, and the Mumbai procured it from outside the country. So then, of course, it's become now a household thing, and now you have so many other generations later on. And I think what is Professor Gupta was rightly expressing his concern about the issue of this drug resistance. So I think that is something which requires tremendous amount of public awareness and the tendency of avoiding self-treatment. But more than that, what is often not said is something which pertains to my own community. As Francis Bacon once said in the context of English language, he said the greatest harm to the English literature is done by the teachers of English language, by asking their students to sit in dingy classrooms and mug up what is told to them. So they recite Shakespeare by heart without understanding what it's all about. So much of harm has been done by my community of physicians who actually prescribe antibiotics left and right. And I'm sure Dr. Y. K. Gupta, though not being a physician, but as a pharmacologist will bear out, though he may be too polite to say that because we are his colleagues from the next department. But much of this wrong which he is pointing out has been done by the community of physicians. And more so when you are in the private sector, because I have had the advantage of working in public sector, working in private sector, working in the corporate world, working in advisories. So I know the gun and avguns of everywhere. The problem happens in the private sector. If I allow the patient, realizing very much that he is on a viral fever, to keep patience and just watch for it or use his thermometer twice or thrice a day, he might get impatient after 3 days. And what happens is that he changes the doctor and I lose the patient. So that constraint of that competitive private practice is also a huge, huge pressure on practitioners like us. And second, of course, is the pressure from the pharma world, because then the OCARD the rep would be sitting outside my clinic knocking at my door even if I finish up my patients at 10 o'clock night, asking me, why are you not prescribing an Nafithromycin when we hosted such a big seminar for you? So all these, so unless we realistically address these issues, which of course will take time because it's also got bearing with a lot of our socio-economic dynamics, it will take some time. But at least we have started talking about it. And I'm glad that Dr. Professor Gupta has very nicely flagged out this issue, because otherwise the day is not far when you'll have to look for something else than Nafithromycin. And Nafithromycin is, of course, most of the trials conducted in the resistant respiratory infection, but has a bearing on also the area that I practice in, because most of the diabetic patients are prone to have the resistant infections, including the respiratory infections. And once, God forbid, he has an aspiration, the pneumonia becomes really very tough to treat, and the immunity is already compromised, and one thing leads to the other. And very soon, if you are not guarded enough and the sugar levels also start fluctuating, the patient may suddenly sink into septicemia. So this is one drug which will come handy to my genre of practitioners as well. But again, we'll have to guard against this, that we don't again make it redundant and start looking for some next generation to overcome the resistance to this drug also. So that note of caution is something that I think all of us, whether we are in the pharma sector, whether we are the medical practice, or we are in the pharmacology, or we are in the biotechnology, we need to make each of the stakeholders aware, because it's not only for a concern of the scientific fraternity, it is a concern of the common citizen as well. And therefore, I think we have made a beginning, and as Jitendra Kumar was rightly mentioning, the first indigenous antibiotic against resistant infections from India is also in keeping with the spirit of Atmanirbhar Bharat and Make in India. So we have made in India something not only for Indians, but also who are non-Indians. So that's a huge contribution. I must compliment you once again. Thank you very much.
Thank you, sir, for your kind address and your continued support. We are certain that your continued guidance will inspire many more such success stories in times to come. The House is now open for interaction of the Honorable Minister with press. Any questions, please? So if there are no questions, yeah. So as a gesture of our gratitude, we would like to present a small memento to the esteemed dignitaries. May I request Secretary DBT Dr. Rajesh S. Gokhale to felicitate Honorable Minister with a memento. May I request MD Bairak to present a memento to Secretary DBT Dr. Rajesh S. Gokhale. May I request MD Bairak to please present a memento to Professor Y.K. Gupta. May I request Dr. Anamika Gambhir to present a memento to Dr. Habil Khurakiwala. I once again request Dr. Anamika to present a memento to MD Bairak. Thank you, ma'am. So proceeding with the vote of thanks, it is my absolute honor to present the vote of thanks on behalf of Department of Biotechnology and BIRAC. First and foremost, we'd like to express our heartfelt gratitude to the Honorable Minister Dr. Jitendra Singh Ji for gracing this occasion with his esteemed presence and for his unwavering support in all our endeavors. We extend our sincere thanks to Secretary DBT Dr. Rajesh S. Gokhale for his unflinching support, invaluable mentorship, and constant encouragement. Thank you, sir. Our acclamation is due to MD Bairak, Dr. Jitendra Kumar, for his guidance in extending all the support in organization of this program. Dr. Habeel Khurakiwala, Founder Chairman WOCAD, we deeply appreciate you taking the time out of your busy schedule, sir. We thank the esteemed speakers for the day to have agreed to deliver their talks on a very short notice. We would like to thank Mr. Vishwajit Sahai, Additional Secretary and Financial Advisor, DBT, and Ms. Ekta Vishnoi, Joint Secretary, DBT, for their fervent and unstinted support in all department activities. A heartfelt thanks to Dr. Alka Sharma and Dr. Sanjay Mishra, Senior Advisor, DBT, for their constant guidance. My heartfelt gratitude to Dr. Anamika Gambhir, Advisor DBT, for standing tall to the entire event organization, having faith and instilling confidence to do things. My due regards and thanks to Dr. Saxena, Head Investment and General Manager BIRAC, who's also been heading the BIPP program and has been instrumental in putting this program together. Thank you, Sonali, for those multiple iterations. I sincerely thank all the members of the audience, offline and online, for sparing time and marking your presence on this important occasion. Last but not the least, I'd like to thank the senior officials of the department, the entire DBT and BIRAC family for their untiring support in organizing this event on a very short notice. Thank you so much, everyone. We invite you for a lunch outside.
On a short notice.